conference, and very welcome. So I'd like just to spend some words about yesterday. So we got more than 300 participants, and then more than 3,500 visualization. So it was the first day was very successful in terms of what we expected. So just today is the full day. Also, we will touch particularly two different sections: one on prevention in the field and intervention strategies. So we have also, as you know, the break hours to 1 p.m. and then so we have also an introduction out of we have a speaker from EFSA, the Jean Moore, that we are to take in advance. So now I'd like to give the words to Antonio for his excellent introduction.
Good morning, everybody. And so let's start. Good morning, everybody. Let's start soon with the program of the conference. We have as a first speaker for the session Prevention in the Field, Susan Vogelsang from Switzerland. She is the leader of the work package for mycotoxin prevention strategies, and she has a talk titled Mycotoxin Prevention Strategies, Main Outcomes from the MycoKey project. And I like to give the words to Susan, and oh, there is a technical problem. She is not online, but we send maybe we can start with Dimitrios Dracopoulos waiting. Susan Vogelsang join us in the conference. Sorry for this technical problem. So Dimitrios Dracopoulos from Switzerland, he will talk about innovative cropping system and biopesticides to prevent mycotoxins in wheat. Thank you, Dimitrios. You can start the conference today.
Thank you very much. Good morning. So, yeah, I just share my screen.
Yes. Everything is fine.
Okay.
Go ahead.
Okay, so good morning, everybody. I will present you today a big part of my PhD work. I did that at Agroscope in Switzerland, and I was supervised by Susanne Vogelsang and also Professor Johann Six from the group of Sustainable Agroecosystems at ETH Zurich. And I will talk to you about innovative cropping system and biopesticide to prevent mycotoxins in the field. So a brief introduction of the life cycle of Fusarium graminearum. Here you can see a field after it was harvested maize crop, and this is the best environment for Fusarium graminearum to establish. So you can see here the maize residues. This is a very good environment for the inoculum to develop. And later in spring, we have the ascospores, which are discharged and they infect the wheat heads. And also we have the asexual spores, the conidia, which infect also the wheat heads and they create the disease. And as we all know, we have big yield and economic losses, but the most important factor of Fusarium head blight is that they contaminate the cereals and make them unhealthy for us. This is to show you a very interesting meta-analysis. So here we see that with the best available fungicide, we only have a reduction of disease and reduction of mycotoxins up to 40 to 50%. And therefore, there should be some alternative solutions to control Fusarium head blight without the use of chemical pesticides, fungicides in this case. So there is an increased pressure when we continuously cultivate cereal after cereal in the crop rotation. So, for example, mate-wheat rotation is still common practice, but this is the most favorable environment for Fusarium head blight disease. Also, when we have no tillage system or reduced tillage system, you can see here in this photo, we have a lot of crop residues left on the soil surface, and this is a very good for the soil quality in terms of soil quality because we have reduced soil degradation and erosion risks.
Devi passare tu. Vai. Unfortunately, we had problem. Unfortunately, we have a problem in the connection. Dimitrios, could you listen us? Today we have problem. It appears that we have problem from connection with Switzerland. I don't know why. Maybe the best is to keep going and to start again with after with Dimitrios and Susan. I would like to invite Lorenzo Stagnati, that we know is in online, waiting for his talk, and we will go back on Susan Vogelsang and Dimitrios Dracopoulos when they will be available online again. Lorenzo, are you ready for your talk?
Good morning to everyone. Can you see my screen?
Not yet.
Hear me?
Okay. Will be soon. You will be soon able to. You can share this.
Can you see the screen?
Yes, please. Okay.
Okay, perfect. Thank you. Good morning to everyone, and thank you to the organizers for the opportunity to share our work entitled Unraveling the Genetic Basis of Fusarium Resistance in Different Maize Populations. Fusarium verticilioides is a widespread pathogen which is found wherever corn is grown. It is able to cause several disease symptoms on maize, especially on maize ears, and it is important because it is a fumonisin producer. Since fumonisin are carcinogenic mycotoxin, their presence is strictly regulated at the European level in grain stocks, and the presence of fumonisin is monitored year by year with huge fluctuation according to different environmental conditions. And last year, 2019, was a very important year for fumonisin presence in Italy because fumonisin were above the threshold level in 81% of samples that were examined in northern Italy. Since the control of Fusarium is of crucial importance, the most effective way to control the presence of this fungus in maize and the fumonisin in grains is the development of a genetically resistant hybrid. To identify QTLs, host genes, and the genotypes that may contribute to resistance to Fusarium verticilioides and to the accumulation of mycotoxin, we use the genetics, genomics, and the transcriptomic approach to test 4 different maize populations. The first one is an association mapping panel. The second one is a biparental population. Then we moved to a multi-parent advanced generation intercrosses, which is also called MAGIC. And at the end, we test a population composed of F1 hybrids we have developed starting from resistant genotypes. The maize core diversity panel is an association mapping panel composed of 302 inbred lines that captures the majority of biodiversity present in public breeding of corn. This panel is composed of temperate tropical inbred lines, as well some sweet corn and popcorn inbred. The lines are available upon request, and the genotypes are publicly available and obtained by GDS sequencing. The parental population are segregating populations that are generated crossing two inbred lines of contrasting phenotypes for the character of interest. In our case, we developed this population crossing the resistant CO441 to the Fusarium yarrot susceptible CO354. After selfing, we generated a progeny of 188 F3 families that were phenotyped in the field and genotyped by the Illumina HiSeq platform with SNPs and microsatellite markers. We have available also RNA-seq data on control and inoculated kernels of both the parental lines. The magic maize population is a structure composed by 8 founder lines selected to maximize the diversity present in maize. Those founder lines are crossed together in a funnel scheme to produce a 2-way, 4-way, and then a 8-way hybrid. And by the selfing of the 8-way hybrid, we collected a population of 1,636 recombinant inbred lines. 1/3 of the population has been characterized with the 50K SNP chip array. We have available RNA-seq data from the 4th leaf of the 8 founders line and the 96 reels, and RNA-seq data on control and the Fusarium inoculated seedling of the 8 founder lines. The collection of resistant hybrids was developed by our university using female lines that were bred here, starting from Italian lines of the LO series that were crossed to United States line of the MP series, because those MP series is known to be resistant to Fusarium and Aspergillus yarrot. As a male lines, we used 3 Canadian lines of the CO series because those are reported to be important sources for resistance to Gibberella yarrot and Fusarium yarrot. Those hybrids were tested for Fusarium yarrot resistance, pulmonis in accumulation, and agronomic traits in Triviano and San Zenone al Lambro in the province of Milan in 2 years, 2017 and 2018. When we select materials for Fusarium resistance, we have the necessity to have some technique to test the resistance. And so in the field, it is possible to test the resistance of the materials with the side needle inoculation method or the toothpick method. We prefer the side needle because it is the most effective to simulate the presence of the disease in our environmental condition, and phenotypes are taken on maize ears at harvest maturity. In a laboratory condition, we developed a bioassay which is called the roll towel assay, which enable us to test the resistance of germinating seed and young seedlings after 7 days of incubation. Independently from the population we have tested and the phenotype method we have used, we were able to see a wide phenotypic variation for Fusarium resistance. Phenotypes are usually distributed according to a normal distribution, which is typical of characters under a polygenic control, with a few genotypes that are highly susceptible or highly resistant. Moving to the result, on the maize association panel, we conducted a genome-wide association study that found a huge number of SNPs that were associated with the different traits we have measured. The most interesting SNPs and the genes that were highlighted by this analysis are located on chromosome 3 inside a cytochrome P450, on chromosome 4 inside a chitinase V1, and on chromosome 7 inside a working transcription factor 125. On the biparental population, we ran QTL mapping. We were able to identify several QTLs that were associated to Fusarium yarrot, to fumonisin accumulation resistance, or to both traits. The most interesting QTLs are those that are common between Fusarium yarrot and fumonisin accumulation. And inside the QTL, we were able to find some genes that are putatively involved in defense mechanism in plants. On the MAGIC population, we found 3 QTLs, 2 on chromosome 4 and 1 on chromosome 5. Inside each QTL, we ran association mapping to identify candidate SNPs, and those candidate SNPs were then compared to the genes that were highlighted by the differential expression experiment on the 96 reals and 8 founder lines. And with this kind of combined approach, we were able to find a good candidate in a senescence associated protein inside the QTL 4.1. The resistant hybrid collection shows interesting differences between hybrids and location we have tested. Inside this kind of collection, we were able to identify a group of female lines that are part of the S5 group as the most suitable to combine resistance. On the male contributors, we found that CO441 is the most promising source to transfer Fusarium yarrot resistance. We found good correlation between Fusarium yarrot and fumonisin accumulation in both localities we have tested. And as an agronomical point of view, we found that yield potential is conferred by the S5 female group, as well as from CO441 and the CO433 as male donors of pollen. This study allows us to identify 5 S5 inbred lines that are now proposed for plant registration at CPVO. We are going forward, and so we produced a collection of hybrids starting from the MAGIC population. We used 430 reels, and we produced more than 500 RICs that are those kind of hybrids using this kind of crossing chain. So the hybrids were tested with the roll towel assay to assess that the hybrid population have a good resistance compared than the collection of the reels. And so this suggests a possible effect of heterosis for Fusarium resistance in the seedlings. Heterosis is very important in commercial maize, and it is poorly investigated for Fusarium yarrot. And so we are moving to this kind of approach. Since we have the genome of the reel, we were able to reconstruct the genome of the RICs using the 2 genomes of the reels that produce each RICs. We tested that the population of the RICs is balanced, there are no structural problems, and this is ideal to conduce a genetic experiment. And then we were able to map a preliminary QTL on chromosome 3 on the RICs collection. We are now looking inside this QTL to identify candidate gene for resistance. The same collection of RICs was evaluated in the field here in Piacenza in 2 years, 2018 and 2019, for Fusarium yarrot resistance and agronomic traits. For phenotyping, we tested 2 different approaches, one visual scoring method and one digital method using a particular software to capture the image of each ear. And we were able to find a good correlation between the 2 phenotyping methods. As an agronomic point of view, we were able to see that there is a good group of hybrids here in the red circle that are highly producing and and characterized by low levels of Fusarium yarrot. And this kind of hybrid is a good group to be commercially exploited. In conclusion, the high-density genotyping data that are available on different maize population makes them handy tools to be combined with the phenotyping for the identification of candidate genes for resistance. Many of the candidate genes can be used as molecular markers for marker-assisted breeding for Fusarium ear-rot resistance or Fumonism accumulation. And from those different approaches, we were able to identify at least 25 genotypes that have been selected, and the most promising 5 are now undergoing for plant registration. Thank you for your attention. If you have any questions, I will be happy to answer.
Thank you very much, Lorenzo. It's very interesting talk, because we know that it's extremely important to develop resistance to this Fumonism producing species, as we know, in the perspective of reducing also in Europe the use of GMO. That is the only way now to control Fumonism producing species. This aspect is very important. But I have two questions related to your work. The first one is well known that Fusarium can attack the maize at 3 different levels, from the seeds, at the silk emission, and through the insect that act as vectors and then can cause holes in the mature ears. Then how efficient can be this kind of resistance toward the 3 different kind of ways that Fusarium use for colonize the maize? And the second, could this resistance you develop have could have a wide resistance toward also other Fusarium species that produce Fumonisms, like Fusarium proliferatum?
So the pathways that Fusarium has available to infect the plant are very different, and we are moving in this kind of 2 different testing to have a control on both the ways of Fusarium has available. So the roll towel assay is dedicated to resistance in a young seedling, and then in the field we can use the most resistant after the roll towel assay experiment to find those genotypes that are also resistant according to the thin bar inoculation method, the side needle. In our environment, the silk infection is of side importance because the conditions are not so favorable. In the past, we tested also the silk inoculation method, but here in Piacenza, we were unable to develop this kind of Fusarium disease through this kind of inoculation, and so we abandoned this kind of phenotyping method. The second question, can you repeat, please?
If this kind of resistance can be also efficient against other species that produce Fumonisms, like Fusarium proliferatum, which is an important pathogen of maize as well.
Rob, actually, I don't know, because we have never tested the presence of different fungi. We use one single strain inside the inoculum. Probably the effect may be transferred also to different different species, as reported from published paper. We tested the same genotypes for Fusarium ear-rot and Aspergillum ear-rot resistance, and we were able to identify a group of materials that are resistant both to Fusarium ear-rot and Aspergillus ear-rot. Maybe with inside the same genus of the fungi, we can have more correlation.
Okay, thank you very much. Thank you again for both answers and presentation, and I hope this kind of job will allow us soon to have a fully available tool against these guys that produce these dangerous mycotoxins on maize. So now we solved the technical problem, and we have online Susan Vogelsang from Switzerland. As I said before, she is the leader of Work Package 4 mycotoxin prevention strategies of MycoKey project, and she will talk about the main outcomes from the MycoKey project of this work package. Thank you, Susan. The presentation is now for you.
Thank you. Can you hear me, everybody?
Absolutely, yes.
Oh, perfect. Thank you so much, because I got about 1,000 links to join in listen only mode. And now just 50 minutes ago, I got the one as a panelist. So I apologize for the delay, and it's my pleasure to present some of the main outcomes of Work Package 4. I put on purpose as the cover slide one from our technological workshop that was pre-corona taken, so everybody was close to each other, and we had a great time about how to prevent mycotoxins in the field. Oh, it even works, good. So I give you an overview about the main tasks that we had in this work package. The first one was about how to control Aspergillus flavus with atoxigenic strains. There was first a Europe-China program that was developed, and also work on optimization of the biocontrol agents in Africa. The second task was about how to reduce Fusarium head blight inoculum and mycotoxins in cereals, mainly in cereals, but also in maize. And there was a subtask on what would be the optimal cropping systems, which was actually more or less the same as the second subtask, crop rotation and tillage, and also a very vast subtasks on the effect of undersown crops, cover crop species, and biofumigation approaches. And then there was also a very interesting task on how to control Fusarium graminearum with biocontrol agents in contrast to other approaches. This was on how to apply them not on the main crop, but on the previous crop residues. And the last task is about resistant efforts, both in maize and in wheat, and to evaluate also durum wheat varieties, how they work in terms of pasta production. Okay, so, and all that was, or two of those tasks were also accompanied by economic feasibility studies, because it's not enough to find approaches how to control Fusarium head blight, but is it something that's worthwhile to be used by the grower? Is it too expensive, or what are the main obstacles to that these approaches are going to be put into practice? Okay, and with respect to the partners, we had 3 organizations from Italy, Piacenza, Bari, and Syngenta, Italy, then 5 institutes from China, Serbia, Romania, Nigeria, Belgium, and Switzerland. Are you still hearing me? It's so funny if you don't see anybody.
Yeah.
Everything's okay?
Yeah, okay.
Okay, thanks. So I start with the first subtask. What has been done is that in Romania and Serbia, there were lots of Aspergillus isolates that were then sent to Italy for identification using the beta-tubulin gene region sequence analysis. And after identification, there was also a molecular characterization by 16 primer pairs of the aflatoxin Aspergillus flavus gene cluster. And using a microsatellite population analysis, they established the UPGMA tree for Romanian isolates. And what you see, all those marked with a red star, those were the selected atoxigenic strains. And they were first tested in vitro about the efficacy. And what you can see here is in this graph, that's a % reduction in aflatoxin B1. And out of that, the strain 36.3 was chosen. This was chosen because in the in vitro test it had over 95% aflatoxin reduction. So that candidate was then tested in the field in Romania. The problem was that the overall aflatoxin level was so low that we could not confirm the efficacy in the field. So that definitely needs to be repeated, which is always like the nightmare of a plant pathologist. If you don't have any infection, you can't see any differences of your treatments. Then in China, there was something similar going on. They already had a lot of atoxigenic strains. They tested them in Petri plate dishes, in chromatography assays, and what you see here they also did PCR assays to define which are the ones that are toxigenic and atoxigenic. They tested them in the field, in groundnuts and in maize, and what you can see is that there was this AF51 had remarkable aflatoxin B1 reduction ranging between 61 and 100%. What was also done at the same university was they looked at different, or how to optimize the fermentation procedure. You can see that on the picture. That's quite impressive, like large scale fermentation on those kernels. That was something that Sang Hao yesterday mentioned already, that they received an approval to do an experiment in order to go towards a new registration of this biofungicide. Then going on to Africa, there were 2 new aflatoxin biocontrol products developed from Nigeria for Ghana. And both of the products were tested throughout 2 years in 400 maize and groundnut fields. And this is indeed the most consistent and the highest aflatoxin reductions ever observed. Just to demonstrate that, you see here a table with 5, on the left side, you see 5 different locations. The control is the one where no atoxigenic strain was applied, and treated is with the atoxigenic strains, and you see efficiencies between 96 and 100%. So highly consistent and extremely satisfying results. That was also demonstrated then in publications about the aflatoxin prevalence, about biocontrol strain identification, and fungal community characterization. So both products, they were registered with the Ghana Environmental Protection Agency. They observed, what you see on the left side, a 3-year registration. IATA is currently discussing with an interested company about technology transfer and respective licensing agreement. And they launched initiatives for the public and the private sector to help upscale the production of those AflaSafe products. And on top of it, the Ghana Standards Authority, they launched a national aflatoxin sensitization and management initiative to increase awareness for both, like about the importance and the danger of aflatoxins, but also about the possible solution, an AflaSafe product across the entire country. And this has to be mentioned, was not only funded by MycoKey, but several funds like the US Africa International Development, the Africa Rising Project, and from the USDA. So I'm moving on now from Aspergillus to Fusarium head blight. I combine on purpose the subtasks 2.1 and 2.2 because one was on the cropping systems and the cropping factors, and the other one about crop rotation and tillage, which is sort of interchanging. So what I'm first presenting is a long-term study that has been done in Belgium about deoxynivalenol in wheat, and about those 18-year survey. What you see here is the DON content on average with the worst case scenario. That means you have maize as a previous crop, susceptible cultivar, and you do not plow. So you leave the residues on the ground. And then they compared that with other scenarios and see about how the DON content was reduced. And the best option was to simply not use maize as a previous crop. And it's said simply, it's still something that's very common in Europe. And Dimitrios is going to talk about that later on a little bit more. In parallel to that, we did in total 4 years, but within MycoKey, a 2-year survey in Bari. And there we observed a quite similar pattern. On top of it, it was also observed that if you do use strobilurin-based fungicide, you actually get an increase in DON, which is explained by a greening effect and also wiping out all kinds of other pathogens and leaving room for Fusarium graminearum to develop. Then going on to the next subtask, which is about undersown and cover crop species, and in this case, biofumigation. That was done in Belgium. What you see here is the results of a petri dish method where they looked at all kinds of different isothiocyanates from different Brassicaceae species. And you see, like with the Brassica junquea, the Indian mustard on the right side, there's no growth at all from Fusarium graminearum. That is exemplified by this graph. What you see here is the reduction of mycelium growth from different Brassicaceae species and different varieties of the same species. Quite nice, while rather low reductions from those species that I show here. But in contrast to that, the Indian mustard, we had a reduction between 53 up to 98% of Fusarium graminearum growth. And this was also confirmed in a field trial. You see here on the right side, that's the Indian mustard. It was put back those species into a pot experiment, and then you saw the same effect that Brassica junquea was the one that was most efficient. Okay, then something similar was done in Serbia. They looked at the effect of white mustard on DON and zearalenone contents in barley. What you see here, that's a different kind of graph. On the left side, you see results from plots that were not receiving infected maize stalks. And on the right side, you see those where it was either having white mustard as a cover crop or not. And those with the triangle are that's the DON content where white mustard was applied. And overall, there was a DON reduction between 75 and 85%. And nicely enough, the same effect was observed for zearalenone. So that's kind of nice that you see that also in different areas within Europe. Okay, and that's a talk of Dimitrios Zakopoulos, so please stay tuned. You're going to be in for a nice treat. He's talking about all kinds of different intercropping, cover cropping, and biofumigation experiments in wheat. And now I move on to the biocontrol approach. As I said in the very beginning, this is a different approach than most commonly used. You do not apply the biocontrol agents directly on the wheat heads, but the fall before on previous crop residues. And what I'm going to present now, has been done in CNR about the Trichoderma atoburneum strain, the item name 901. This is owned by CNR. It's an authorized active substance, and for horticultural crops, and I believe it was against apple scab in orchards, this is almost completed, the registration procedure, and it's close to the marketing phase. So the question was, within MycoKey, could that also be used against Fusarium graminearum in wheat? So what has been done was first doing in vitro studies in Bari with 20 different trichoderma isolates, and then using the item 908, what could be done in the field. So in the lab, they looked at 20 different strains of trichoderma with simple confrontation cultures. And what you can see here on the top are perithecia on carrot agar. And the interesting thing is, what's easy is if you have something that's pre-colonized by the biocontrol agent, like in trichoderma, 80 to 100%. We have also observed that for many, many different biocontrol agents. But the most important one is if you have Fusarium graminearum, the pathogen, in the very beginning, and then you have it being confronted by trichoderma, and there were still 5 strains that inhibited the perithecia production by over 90%. So what they also did, they did sequence the genome of 908 and looked at the time course of 8 different genes that were expressed in Fusarium graminearum during the formation of the fruiting bodies of the perithecia. And this genome will be used in the future for studies like what's the mode of action of trichoderma, which isolates could be promising, and could we get some markers that could be used in studies on population dynamics of this species. So this has been tested in the field in Switzerland. Alejandro Jimeno, which also, like Dimitrios, successfully defended his thesis. He did some field experiments. What you see on the left is the approach. So what we did was collecting maize stalks from the field, autoclave them, and then inoculate them artificially with known strains of Fusarium graminearum. And after that, added the biocontrol agents. And after incubation, then we put them back out to the field. You see that here in the middle picture, where you see wheat plots with the treated maize stalks. And we used a split plot design with 2 different winter wheat varieties, Levis and Forals, with different susceptibility and ranges and flowering time. So the treatments that we had was, in the beginning, was a strain from the Netherlands, and Clonostachys rosea, I'm sorry, and this item 908 strain from Italy. And we had 3 different treatments. One was the unformulated cornelia, just washed off either from the grains from trichoderma or from Petri plate. Then we did have a vegetable powder product where the strain of Clonostachys was produced on barley grains. And another one was a formulated product where we, apart from oil, we also use UVB protectant agent because Clonostachys rosea is a soil-borne pathogen, is very sensitive to UVB radiation. So the results for that was, first of all, what you see on the right side, those hatch bars, those are the blank formulations without any agent. On the left side, on the very left, you see the negative control, which means maize stalks without any Fusarium graminearum on it. On the red row, those are the ones where you had Fusarium graminearum and the DON content, the respective DON content. The yellow bars are all the different Clonostachys rosea treatments, and the green bars are those, the DON contents when you treat it with trichoderma. It was nice to see that because Clonostachys rosea in both years on average reduced the DON content by up to 93% and zearalenone by 98%. Interestingly, in the first year, we didn't have any effect from the trichoderma isolate, but in the second year, which was more humid and warm weather conditions, we also had really nice reductions of DON by up to 90% and zearalenone up to 87%. And this has been published previously in the Journal of Applied Microbiology. Okay, so this has also been tested in Serbia, trichoderma, the 908 strain. And same approach for the graph. On the left side, you see the negative control, like no Fusarium graminearum on the stalks. And on the right side, you see treatments either with no trichoderma, those are the squares, and those on the circles on the bottom side, those are the ones that received trichoderma treatment. And we did get remarkable reductions up to 80% for DON and even over 80% for the zearalenone reduction. Okay, so as I mentioned in the beginning, it doesn't help if you have wonderful cropping systems or working biocontrol agents if they're too expensive or too complicated to be used into practice. So what they did, they compared the costs with the treatment effects. And the indication was just for the Synapsis alba treatment, to only decrease the toxin levels, that would not be enough. What's needed here is also to look at the other benefits. because if you do have a cover crop, you have also less weeds and nematodes, and on the long term, you have more organic matter in your soil. So what we believe we would need adjusted agro policies and also training, of course, to farmers to promote this kind of cropping system. For biocontrol agent, it was the same thing. For now, it is too expensive, the production of those biocontrol agents. So also that would need to be supported by direct payments, for example, in order to foster the development of biocontrol agent products in cereals. Okay, I'm moving on now to the resistance task. So that was done in Italy, the maize breeding field test where Lorenzo was already talking about. They identified 25 less susceptible inbred lines in 3 maize panels of different origins. They developed new experimental hybrids and they tested them in the field against Fusarium erod. They did that throughout 2 years in 2 sites. You see here an example of a maize nursery, and from that on they selected 5 inbred lines that were really well performing in the field. You see here on the left side maize cobs that didn't show any symptoms or very few symptoms, and they were recommended for registration in Italy. They also looked at different genetic markers for Fusarium erod and seedling rot in maize. And what you see here, the genetic material, there was a biparental population. They identified 2 QTLs which are involved in Fusarium erod resistance and resistance also to fumonisin accumulation. 3 QTLs which were linked to the resistance to Fusarium seedling rot, and also 3 SNP markers also involved with Fusarium seedling rot resistance. Something similar was also done in China. They did a genome-wide analysis, genome-wide association study, and identified new QTLs linked to resistance, looking at the Fusarium head blight severity, the DON content, and the best linear unbiased prediction indices. And they identified 6 new resistance QTLs, and patents in China were filed for those QTLs. Then they were also looking for resistant, or let's say less susceptible wheat varieties in different locations in China, looking at different varieties. And they found, in fact, 10 moderately resistant wheat varieties with a down reduction between 7 up to 78% compared with the main cultivar or the most common wheat variety. One of them is already in the market, and others are still in the registration procedure. Then done by Syngenta and CNR, they were looking at durum wheat varieties for pasta production and whether they are less susceptible for Fusarium head blight and still comply to the quality parameters that you need. So they were using a phenotypical approach to select the most Fusarium head blight tolerant varieties, looking at also at the economical and quality profiles. And the way this was done, they had a field experiment over 4 years at 2 sites in Italy, and they had both, like a natural infection as well as artificial inoculation with known Fusarium graminearum strains. Oh, I think it was Colmorum, sorry. They tested 10 different varieties. So standard varieties that are known in the market and used quite a bit, and susceptible ones and less susceptible ones. And then they looked at the symptoms and the DON content. And the goal was to provide the farmers with new varieties that show both a satisfying performance in terms of yield and all other quality parameters, as well as low DON content. And that the ultimate goal is to provide, of course, raw matter, which is of highest quality and low toxin level to the industry, and eventually, of course, to the consumer eating pasta with an improved food safety, and no compromise on the food quality. So these are the results or selected results from the field experiments. What you see on the left is the symptoms from Fusarium head blight, either on not inoculated sites or at artificially inoculated sites, and on the right side, the down content. And you don't have to go into detail that. What they did found was consistently that the variety Efeso and Atlante, they had the best combination of, or the best balance within yield quality parameters and also low down content under different disease levels. And that led to a quite nice recommendation to farmers about the varieties that could be used and that are sufficient for the pasta downstream needs. And these results are, of course, also relevant for breeders about which parental line to be used for further processing to create new improved varieties. Okay, so I'm summing up. The conclusions were that we did find excellent atoxigenic Aspergillus flavus strains. 2 products are on the market, AflaSafe GH01 and GH02. And 1 is under registration in China, and that gives good prospects for safe ground nuts and maize products. For Europe, we do have atoxigenic strains, but since the aflatoxin levels overall were so low, those field experiments would need to be repeated to confirm their efficiency. We did evaluate cropping factors that decrease the risk of Fusarium head blight in both in wheat and barley. And we found cropping systems, different intercrop systems, cover crops, and biofumigation approaches that substantially decrease both the DON and the zearalenone contents. And we believe that we do need adjusted agro policies to promote those promising systems. Both Stachys rosea and Trichoderma atroboneum are promising biocontrol agents, and in Switzerland, in fact, the current efforts are towards a microencapsulation approach to further improve the survival and efficiency in the field if you apply that directly on the maize residues during maize harvest. And finally, we found several less susceptible maize hybrids against that are tolerant against Fusarium ear blight. And wheat varieties that are less susceptible for Fusarium head blight. And identification efforts were quite successfully in identifying several markers like QTLs, SNPs, and also metabolites. So I would like to thank you, all the work package 4 partners that were providing the reports, their latest results. I would also like to thank all the many undergraduate and graduate students and interns helping the work across Europe. Of course, the participating growers that were sending us their samples and cropping factors, your attention, and it would be nice to discuss as we did last year in Zurich, close to each other. But if there's still time, we can discuss virtually if you have any questions. Thank you very much.
Thank you, Susan. I'm really impressed by the amount of work you have coordinated in this work package, and extremely efficient and high quality research. I have a question.
Yes.
In the perspective of a Green Deal that will be the base of the next European research program, Horizon Europe, how far we are, in your opinion, in using all tools that will avoid use chemicals for avoiding the Fusarium contamination on Fusarium in Fusarium blight of wheat, for example, or maize. How do you think we are, our research are advanced for getting this aim?
That's a very good question. I think you're talking about Fusarium, right? Not about Aspergillus right now.
Well, Aspergillus, I see that we have really
We do have
excellent and
programs.
promising result with this atoxygenic strains.
Yeah, yeah.
Still on wheat and on fusarium reduction by using biological control, I think we need maybe some work, but I would like to know, in your opinion, how advanced we are at the moment.
Yeah, I think for fusarium head blight, it definitely needs a combination. Like, it doesn't help if you have an excellent biocontrol agent, but you still continue to grow high yield but highly susceptible wheat and barley varieties. I think it really needs a combination of less susceptible varieties and then adding another approach. As I mentioned, the biocontrol products, so far nothing is registered in Europe. And I think a lot of efforts have to be done for formulation, which is true for both, for the biocontrol agents, but also like for botanicals that are working quite well in a lab and a climate chamber experiment, but are not protected from washing off or from UV radiation being destroyed. And the key, I think, is getting decision makers involved so that the agropolitics help in getting farmers to use those cropping systems, for example. So making them aware, see, we do have, for example, let's say what Dimitris will mention later on, we do have an excellent working cut and carry biofumigation or a cover crop system. We need direct payments for the growers in order to use them. And then I think we're a great step towards having cereal products with a low DON and zearalenone content. I think we're pretty close. It's the last step that's the most difficult one to get agropolitics involved.
Thank you. This is a really exhaustive answer. I really appreciate it. Thank you very much, Susan, for excellent presentation, the nice answer. And I move forward. We are able to. Bye. We are able to call again Dimitris Dracopoulos. Sorry for the interruption before. He will speak again about innovative cropping systems and biopesticides to prevent mycotoxins in wheat. Dimitrios, you can start to talk. Still absent? Okay, I'm afraid that we have still a technical problem, so I would ask to the next speaker. No, he's there. Is there? Unfortunately, there is a problem with the microphone. So I think we should move to next speaker, who is Ranajid Bandyopadhyay from Nigeria. Ranajid will talk about reducing aflatoxin contamination in several African nations through use of special atoxygenic biological control products. And Ranajit, please, you can share your screen.
I need to find out how to share.
Yeah, you share it. We can see your presentation already. No.
You can see my presentation?
Yeah, before we could. Now, not. But you were able to share it before.
Okay, now the issue is that if I share it, then
okay, you share it now. You share it now. Yes, you can go.
I can't see my presentation now.
We can see it.
Can you see now?
Yeah, sure.
Oh, okay, great. Thank you, Antonio. And it's really a pleasure to to talk to this at this international conference. I particularly feel privileged because I have been participating in the mycotoxin projects, new mycotoxin projects since the last 3 projects, right from MycoGlobe and then to MycoRed and finally to MycoKey. And as I mentioned, I'm really fortunate in that. What I would like to speak is about our work on biocontrol in Africa. Now, this is, and I'm presenting on behalf of the IITA AFRISAFE team. There are many members of this team. I'll show the names later on in the pictures. First of all, I know this is a mycotoxin conference, so all of you know what mycotoxins are. But just to remind that the situation is critically grim in Africa, and it transcends through the public health, it transcends through trade and economy, and food and nutrition security. About 30% of all liver cancer cases in Africa, they are attributed to the aflatoxin. About 40% of the commodities in local African markets exceed the limit of aflatoxin in foods. And they estimate that Africa loses $100 million or more annually from lost potential export. Aflatoxin actually gets into the newspaper headlines quite often, and you'll see some of the recent, this is actually in the last 1 year itself, just a sample of various newspaper cuttings that we actually see when aflatoxin events take over and people become very aware of it. I don't know what's happening.
You should go forward from the other.
Yeah.
Okay.
So if you actually look at it right from planting to consumption, there are multiple practices that can be followed for mitigating aflatoxin, right from drought-tolerant varieties right up to diet diversity. There are many practices that can be followed. And the ones that I'm actually highlighted in pink rectangles, these are the ones I consider that can be practiced on a larger scale and can reduce aflatoxin contamination to a larger extent. Diet diversity also can reduce aflatoxin exposure. Sometimes it actually gets difficult to implement it. But what I'm going to talk about is about biocontrol. And biocontrol is just a simple field intervention that can effectively reduce aflatoxin contamination from right in the farm until the grain is consumed. Now, let me also remind you of something I forgot to mention, that all the control options that you have here on the top, these are all technical options. And they must be on a foundation of good awareness about institutions and policies. These 3 must support the implementation of the technologies. Now, biocontrol actually works, and in the US, where this was the inoculant strain-based biocontrol, that's what I'm going to talk about. In the US, this technology is now used in hundreds and thousands of acres on several crops, and they're treated with 2 EPA-approved products, right from maize, peanuts, cotton, pistachios, and figs and all that. In Africa, this has been a partnership between IITA, USDA, ARS, and many public and private sector organizations. It works in Africa because until now, various African farmers, they have been able to treat more than 360,000 hectares, and about 250,000 farmers have used it, and they have produced more than 600,000 tons of aflatoxin safe maize environment. So it is practically that can be used, but a lot of effort is required to make it practical and to make it scalable. Okay. Now, to give a very short background about where we come from, Aspergillus flavus is actually always present in the crop, and the crop is already associated. And from the crop, it moves onto the crops. And in the crops, you can get right from 1 CFU, colony-forming unit per gram, to more than 1 million sometimes colony-forming unit per gram. And that results in anywhere between 0 to more than 48,000 parts per billion of aflatoxin. Anywhere between 0 to 20 is safe. Anywhere between 21 to above, they are they are toxic. Particularly when you have levels like 48,000, people can fall actually dead. Even cattle can fall dead really quickly. Now, endoxygenic fungi exists in all the regions of the crop and can be used as biocontrol agents. What we normally do is in each country that we work in, we actually isolate about 5,000 strains from each country, and we screen them through an elaborate process. First of all, we look for those strains that do not produce aflatoxin, and then the non-toxin producing strains are further categorized into various VCG or what we call it SSR groups. We are looking for those groups that have got wide geographic distribution and no toxigenic members. Now, we also do what we call as CAS analysis to determine, to ensure that the strains that we are looking for should have at least 2 aflatoxin and 3 genes with a defective. In culture, they should be able to outcompete toxigenic strains. So right from 5,000 strains, we actually narrow it down to 8 to 12 strains that we select for field tests. These are being applied to the field, and then we look for those VCGs that have superior capacity to colonize, multiply, and survive in soil. They can be frequently isolated, and they have good capacity to reduce aflatoxin. And based on that, we select 4 strains, and we constitute a multi-strain product that in Africa, we call it Aflasafe. Basically, Aflasafe is the trade name for the biocontrol product in Africa. Now, if you look at how we constitute the product, so as I mentioned, we select highly adapted and widely distributed competitive strains. These 4 constitute an Aflasafe product. So these strains are individually grown on solvent grain in bottles and for about 7 days, and we get large quantities of Aspergillus growing on these grains. These are then suspended in water, and then together with a polymer and a dye, this suspension is coated onto dead solvent grains. And the coating is done in a normal seed coater, and it can be done really quickly. I mean, this seed filter can coat about 5 tons of Aflasafe of grains per hour. And then those grains are then taken and bagged. This is the bagger actually, and then bagged and put into the bag. And that's where you have the final product. Now, this product is used at 10 kg per hectare about 2 to 3 weeks before flowering. And there's only one application done in one season. And it usually costs in various countries in between $15 to $20 per hectare. And once it is applied, the product, the endogenous strains that are put on the product, they begin to stimulate on the carrier surface itself. And the soluble grain actually acts as a food source as well as the carrier. Now, in the non-treated field, we have large quantities. The red surface means toxin-producing strains, and the green circles are non-toxin producing ones. So in a normal situation, in non-treated field, you may have fields or strains that are highly toxin producing that are present more frequently. But in an Aflasafe treated field or a biocontrol treated field, the proportion of non-toxin producing strains that are here, the strains that we apply, those increase dramatically. And since now in the field you have a population of non-toxin producing strains, that results in reduction in the aflatoxin contamination in the crop. Now, Susan has shown this slide, and so I will not actually go into it, but even as she mentioned, we get really very high levels of aflatoxin reduction. This is the product in Ghana that was developed together with MycoKey. Now, the reason why you will get this reduction is because we get displacement of the toxin producers. Now, here you have the active ingredient recovery that we actually get from different sources, different substrates. And the substrates, we actually looked at the frequency of occurrence of the strains that we applied in the soil before treatment, soil and harvest, and grain and harvest. The green bar indicates from the treated field, and the red bar is from the untreated field. You notice that even before application of aflosate or the biocontrol agent, the strains are already present in the soil, and that really is because these are all native strains that are normally found in the soil itself. First time, the proportion of non-toxin producing strains will actually increase quite dramatically. And as a result of that, you also get a large portion of the treated grains that have the non-toxin producing strains. And this is the result, this actually results in the reduction in aflatoxin content that we have seen. Okay. Now, once we have the efficacy data for 2 years, and let me also mention all of these trials that we do, the efficacy trials, none of them are done in experiment stations. All the efficacy trials are done in farmers' field under farmers' condition, and treatments are done by farmers. And so what you see is a real-life reduction of aflatoxin in real-life situation. Okay? Now, so after getting the efficacy data, we also collect safety data, which is the toxicological and toxicological data, and with that, we submit a dossier with which we get a registration. So, but before that, let me get to one more efficacy slide, and that is in Kenya. In Kenya, now this is actually aflatoxin PPD less than 4, less than 10, more than 10, and so on and so forth. The green bar is treated, the red bar is controlled or untreated. We notice that the regulatory limit in Kenya is 10 parts per billion. Many samples that came out of the treated fields, less than 10 parts per billion basically, and actually all of them had less than 4 parts per billion. On the other hand, in the control fields, more than about 50% of the crop had more than limit, acceptable limit, and about 25% had more than 100 parts per billion itself. So this product can actually, this technology can reduce aflatoxin to a very large extent. I mentioned that we take this efficacy data and put the safety data and submit the dossier for registration, and that's where we have the registration certificate from Ghana that Susan showed, and these are the labels. And if you notice, the label actually has a green bar, a green strip, and the green strip actually indicates the highest level of safety by the WHO, what is called the new category of safety, basically. Now, there are 17 products, biocontrol products, that have been registered in 12 countries, and out of the 12 countries, in 12 countries, and out of the 12 countries, 10 countries are in Africa. The other two countries are the US, where there are 2 products, and Italy, where there is 1 product. Now, in terms of the active ingredients, totally there are 67 active ingredients that have been registered by the biocontrol authorities. And out of the 67 active ingredients, 64 have been registered in Africa in the 14 products. And these products are registered for use in maize, groundnut, and sorghum. We are trying to expand the label to other crops such as sunflower, sesame, and seed, for example. Some people are asking for it. Okay. Now, where are we in terms of Africa development in Africa? Come to the advanced part where we are later on, but right now we are working in 21 countries in Africa. And this map, this shading actually shows right from the stages from identifying strains to product being tested by farmers, the products ready for registration and products registered. The green bars are where the product is registered, the one that I mentioned earlier. We are testing products in the orange shaded countries, which are actually Mali, Niger, Togo, Benin. It's also going on in Rwanda. There are other countries where it's going to start very quickly. There are manufacturing plants in Senegal, which manufactures for both Senegal and Gambia, one in Nigeria, there's one in Kenya, and there's one in Tanzania, and then there's the ones that are coming up in Mozambique, and there's one that is going to come up in Rwanda. I'll come to that later on. But we have opposite development growth at various stages in various parts of the continent itself. We talked about aflatoxin, the biocontrol. I must mention that biocontrol is not a panacea. So we actually use biocontrol.
Rajid, I'm sorry. Rajid? Ci sono? Rajid?
No, non è non c'è proprio nessuno. Connessione audio deve essere tentativa di connessione.
Che il server dell'istituto?
No, è proprio
Ma anche gli altri invece?
ci sono qui a tutti.
Ora è tornato. Però aspetta, fammi chiedere se si sente.
No, non si sente. No.
Bisognerà... Come non chiudere? Chi li sta?
Vedi, è tornato l'audio.
E riattiverò perché non è...
No, lui si sente, lui sta parlando e... Ascolta. Stefano ti spiega una strana. Malissimo, no, però lo stanno sentendo. Malissimissimo, però vuol dire che lo stanno sentendo.
Come mai non niente?
Ma io non c'ho l'audio. Io non ho l'audio come...
Anche noi non possiamo intervenire.
Come gestore.
Vabbè, lo metto io da qua?
No, non sei quello da qua.
No. Aspetta. Voi, è tu proprio, c'è Mera Ispa, manca. E allora prova a non uscire. C'è una domanda di...
Antonio, am I receiving messages?
Sorry?
Okay, no, I thought I'm receiving messages from you. I can't see the screen, that's okay. So this is the incentivization scheme, and from the incentivization scheme, when we have some successes, the USAID and the GIS Foundation funded the FSA Technology Transfer and Commercialization Project for Super Food in Africa. Now, I'm not going to details of this project, but just give a few highlights of what we actually obtained from this. So from the Ag Research Project, in the 6 years, there were 22 agri-businesses, or what we call the farmer-producer organizations, that work with more than 75,000 smallholder farmers who applied FSA in close to 100,000 hectares. They used about 1,000 tons of grains of FSA, and and the grain loss, 95% of the grain loss, they actually had less than 20 ppb, and more than 90%, and more than 90% had less than 4 ppb. The European Union though. These common producer organizations and the farmers, they earned $3 million from the project incentives and over $5.3 million from the market premium. And the impact evaluation was done by an external agency, which found that for every smallholder farmer, they had a benefit of $318,000. The incorporation of the safe food in their diets increased by 30%. So in other words, there were actually health and income benefits achieved by the farmers by using AfroSafe. Now, as I mentioned, we actually follow a research to delivery pathway, and this actually gives the entire pathway. And until 4, it's the research for delivery pathway, and from 5 to 8 in the commercialization pathway, or partnership with delivery pathway. So what we do is first we conduct a market analysis for AfroSafe. We look at the commercialization options. Then we mobilize private sector, and through the mobilization, we select manufacturing and distribution partners, and at the same time, create awareness. Then together with the distribution partner, manufacturing and distribution partners, we create a market and conduct on-farm demonstrations and awareness raising. And then the investor is backstop to manufacture, distribute, and build awareness to improve the product delivery itself. Now, the EDC project, what it does, is it registers product, it develops and executes the commercialization strategy, and for identifying manufacturing and distribution partners, it transfers the technologies to these partners, and then technically backstops these partners when we start up. To set up the manufacturing plant, for example, and to scale up and to execute the scale up. And they also create the market for AfroSafe's aflatoxin safe crop to stimulate adoption. So this is done only for 1 or 2 years, and the support is given to the manufacturing and distribution partners. And then we actually do back, we exit from the product itself. Now, having said this, I must actually admire the situation that you have in Europe, where you have large numbers of private sector companies that are willing to invest in many of the technologies that come out of research, and particularly biocontrol. In Africa, the situation is very different. We need to seek them out. We need to tell them that there's a business opportunity here, and then we have to show them the business opportunity for them to go forward and take it up as a business option. Okay. Now, in terms of efficacy and commercial use, now these are large-scale uses now. So when I am talking about lots, I'm talking about each of these numbers multiplied by 30 tons. Okay. And so this is from 4 years data, 2014, 15, 17, and 18. I think there's the thing problem here. So again, we actually see large amount of reduction even in the large-scale usage. And in this year, almost 100,000 tons. We actually measured aflatoxin at 100,000 tons. So that's the number of analysis that we actually did. What we also found is that the variance of aflatoxin in these fields, in the treated fields, was almost 1/4 of the variance in the control fields, which means that not only is aflatoxin low in the treated fields, but they are consistently low in the treated fields, and lot-to-lot variation was low. And so the aggregator and the traders who want to come and procure the grains, they will have a larger amount of confidence because the sample variation in terms of aflatoxin is very low. And as all of you know, sample variation for aflatoxin is really extremely high. And if you can somehow reduce that, that can help quite a bit. And then the same thing was observed on the groundnut in Senegal. in the same story. And here, the reduction of invariance is even more dramatic. Okay. And remember, all these are done in farmer's fields, and so you can imagine even the mean aflatoxin levels in the farmer's field, they are extremely high. I mean, there will be some that will be low, there will be some that will be very, very high. Now, what are the key numbers for commercialization? So I'll just go step by step. So there are 3 companies that are now, so the entire commercialization process for aflatoxin in Africa is private sector there. So there are 3 companies that are licensed to manufacture and distribute in 4 countries: Nigeria, Tanzania, Senegal, and Gambia. In 1 country, in Kenya, the government is manufacturing and distributing the product. And there's 1 company each that is distributing aflatoxin in Ghana, Burkina Faso, and Malawi. Now, these are some of the pictures of the aflatoxin plants. This is the plant in Kenya that we built and handed it over to the government. This is the one in Nigeria, which is going to be dismantled, and all the equipment will go to the private sector, and they're going to set up, they're actually setting up their own plant right now. This plant is in Senegal, which is also producing aflatoxin, and there's another plant in Tanzania that is already producing it. So, in terms of manufacturing and distribution, there have been a steady increase from 1 manufacturing and distribution point in Bari, Nigeria, until 2018. So, currently there are 3 operational plants and 1 new factory under construction in Nigeria. And there was 1 distribution point in 2016, and now there are more than 100 points in 7 countries. Now, the uptake of aflatoxin is primarily driven by business-to-business buyers. And so the business-to-business buyers, they consist of aflatoxin functions for private entities and government. In between January 2016 and September 2020, more than 3,600 tons of aflatoxin was sold and covered 363,000 hectares. And 99% of the sale was business-to-business clients. Of this, the public institution represented 40% of the gain itself. Now, even though the availability of the products has expanded significantly, the uptake is still limited. And those who actually work in Africa would know that adoption of technology is slow in Africa. It takes time for something to catch up. And so we expect this is the same thing that's happening with us, and we expect it to catch up actually. And we are gradually seeing that the government procurements are increasing, large processors are coming in, large industries are coming in and showing interest. Now, in terms of the amount produced and sold, we see that in 2020 there has been a reduction, and that is because of COVID-19. There have been some impacts, and I don't think I'm trying to actually go through it, but in the question time, if you ask me, I'll give some examples. Very briefly, the groundnut export sector in Gambia and Nigeria, they're gradually reviving. In Kenya, one of the government projects that used aflatoxin and produced food to provide aflatoxin safe food to 500,000 Kenyans in the drought areas and the food security project. The aflatoxin is now incorporated in the national agriculture investment plans of 6 governments in 6 different countries. There is quite a bit of private sector investment that is coming in, and through these we are seeing more and more job creation as well. Now, some of the key implementation messages that I actually want to give here is that use biodiversity as a key component of an aflatoxin management system. And so these are some of the things. I mean, try properly, store properly. The creation of awareness about aflatoxin management is crucial, and that's where the partnership with various government agencies, the policy institutions is crucial. There needs to be creation and promotion of testing facilities. So we are gradually bringing the testing facilities right to the farm level. Now here, as you see here, this is the work done by Alejandro. He has developed a method by which it's actually it's an innovation of an existing method, but he has taken it to at the farm level. And the aflatoxin analysis is now done together with the farmers and the value chain actors, right at the farm level, where these people, they can actually see the amount of aflatoxin on the grain, because the reader actually shows the amount of aflatoxin that is there in the grain. And they didn't get convinced whether the grain is actually safe to eat or not, or whether the technology is good to use or not. And so this testing facility taking it to the farm and the market, where results can be shown obtained very quickly and be shown to the farmers and the traders. This is actually very crucial. If you want to start about control in any area, look for the aflatoxin conscious market first for starting out. And the premium is going to be important from these sectors. Now, we are gradually seeing more and more interest from the public sector, such as the input supply scheme. For example, in Nigeria, the Central Bank of Nigeria has got an input supply scheme called the Anchor Borrowers Program. And this year, the Anchor Borrowers Program, they have included AflaSafe in the scheme to supply the product along with other inputs to 200,000 groundnut and maize farmers. And that's why the partnership with government policy institutions, the standard facilities and food and safety organizations, those are important, as well as partnership with industry trade bodies. There are certain key scaling and commercialization messages that I want to actually also offer. Number one, public sector innovation in a new market segment must be championed by the public sector. Remember, we are a public sector organization, we're a non-profit organization. So we have to champion that with initial support from the development investors, which is donors, or uptake by the private sector. This initial support is really crucial. Scaling up technologies, scaling up challenges include low awareness, inadequate market infrastructure, and market incentive and poorly enforced policies. Technology development and commercialization of AflaSafe required intervention by a team in a phased manner. So it is not an individual, but it's a team approach, with the initial product development phase led by science professionals, whose involvement decreases with continuous and leadership from business professionals in partnership with engineering and communication professionals grow. Well-planned commercialization strategy followed by careful investor selection and licensing agreements are crucial for successful technology transfer to the private sector. Technology developers require a few years to champion and steward the technology after licensing it from the private sector, while licensees require a few years for technical, commercial, and communication backstopping before they independently manufacture and distribute AflaSafe. Private sector investors see an opportunity to grow their business with commercially viable products. They are interested in the business case, risks, opportunities, product quality, manufacturing requirements, market strategy, and projected return on investment. Marketing strategy for an entirely new product segment addressing a significant but visual public health risk is more than a sales strategy, which requires a strategic collaborative approach. Okay. Antonio, how much time do you have?
Sorry? Raji, what did you say?
How much time do I have?
Yeah, you can go. You are a bit over, but you still can go with your slides. Okay.
Thank you.
If you can speak up.
innovation that came from one of our team members, Alejandro, who is a very key member of the MycoKey project. So when we actually started when the plant in Senegal was being built, the ceiling was on, and the manufacturer had an order of 230 tons. So I have to say, one company gave that order, but the manufacturer, the investor in Senegal was consulting the company. He was behind time, and so he didn't have a lab to produce the inoculum of the master culture of episode. So what we decided to do was to produce what we call as dry inoculum. And dry, so as I showed you earlier, the after-sale manufacturing process uses spool suspension. The spool suspension has to be made in a lab adjacent to the manufacturing plant. If the lab is not there, you cannot actually make the inoculum. So we prepared the inoculum in Nigeria and then dried it and then shipped the dry spores to Senegal. And so this is what the dry spores actually look like. Okay. So these are produced just like normally in bottles. Then they're filtered and through a membrane. It is dried in a dryer. And then they're powdered, the quality control check is done, and then these are then taken to the manufacturer, and so this was taken to the manufacturer, where this was suspended in the liquid form, and so until now, what we have found is the shelf life is actually more than 3 months. The shelf life work is going on. The contamination risk of the inoculum is reduced. It eliminated one laboratory need for the company. And the product is actually very uniform, and the quality is very uniform across countries. This is the new process, though, and we are still refining and optimizing it and for the industrial scale. Now, this is the results of the dry inoculum. So this is in Senegal. These are in 2 different years, I must say, okay? So in 2018, this inoculum came from the garden, so shipped from the garden. And so this inoculum was prepared in Senegal using dry spore formulation. The inner circle is controlled, untreated, and the outer circle is treated. And again, you see that in treated fields, you get really high levels of reduction, okay? And the level of reduction, the high level of reduction is similar to the case of both the conventional formulation and the dry spore formulation. Okay, but again, in untreated, you had the shaded one is the one that actually has more than 20 parts per billion. So you can see there's really high levels of aflatoxin in the untreated one. But in both formulations, the levels actually went down. So the biocontrol development can actually meet 17 of the 18 goals, sustainable development goals. And in summary, aflatoxins are pervasive in Africa. More efforts are required to translate knowledge into actions for benefiting African smallholders. And I think that's the key, that research should not stay in the lab. It needs to be applied where it is needed. And it needs to be, it's needed by the farmers, by the traders, by the businesses, by the policymakers, by the government. So it needs to go down to where it is needed. The aflatoxin management system can dramatically reduce aflatoxin contamination and improve food safety and security. There are institutional and policy innovation must support this technology adoption. And we are currently scaling up certain models for technology adoption. We have several partners in the project, and so these are some of the partners who have been fitting to one screen, both from the private sector side, public sector side, universities, institutions, companies, governments, and so on. We have been fortunate to receive funding from various donors, including MycoKey, through the European Union, and most recently by the French Development Agency, which has funded a project in Sudan, and the ABI, which has funded a project in Uganda. And we have a team that actually goes through from the USDA and IITA that works on greener technology, on product development, field testing, on manufacturing, on advocacy issues, commercialization, business development, communications. We also do work on breeding. We have students in there and project managers, and we have one person each handling socioeconomics and policies. Without the team, I don't think we can, the involvement of the team and the advocacy people was really crucial in the work that we do. Thank you.
Thank you very much, Rajid. This topic has an extreme importance because, as all we know, the mycotoxin and the aflatoxin problem, particularly in Africa, is still a dramatic issue that can cause also death or acute poisoning of people. There is a question from audience, please, Alessandra.
We have a question from Franz Verstraete of the European Commission. To which extent Atlas is used in groundnut production in Africa, and if it is effective? As I understand that Aflagar is not used in the US in groundnut production, as it appears to be too effective.
Right, so there are reasons why Aflagar is used for groundnut, but maybe not to a large extent, and there are reasons for that. And one of the reasons is about the insurance that is there in the US. Sometimes it's cheaper to buy the insurance than invest in the product. Okay, and so that is one of the reasons. Okay. And the second thing is, if after using the product, if you don't meet the standard, then there's a problem. But most often, what I have learned is that the insurance is cheaper than buying insurance is cheaper than buying the product. Because the insurances are not available in Africa. In Africa, what we are seeing is more and more groundnut farmers are actually using it. Just to give an example, the new project in Sudan that we have, the need for the project was expressed by a private company in Sudan that approached the French government to invest for the groundnut value chain. Okay. And similarly, in the Gambia, where it's been used extensively to revise the export market in the Gambia. Just today, I saw a report from Burkina Faso, where the company called Innofaso that is working with Cruscan D. They want to reduce Africa with the groundnut export from Argentina, and they are expanding the use of rapid in Burkina Faso. So we are seeing an example. We're seeing an example in again in the groundnut sector in Nigeria, where the company that has never been able to export to the EU have now begun to actually export to the EU. So we do see examples that is being used in the groundnut sector.
Okay, Rajid, since the audio was not perfect, I would appreciate if you can send us by chat or by mail the last answer you provided in a way that I can reply later when we start after the break or before the break, because unfortunately it was not very clear. So because of audio connection. So please can you summarize what you just answered, and then we will report to the audience. We have to run because we are very late now. So for people of the audience that would like to make questions to Ranajit, we can forward him the questions. And Ranajit, please, you can reply because there is a comment and there is another question, but we really don't have more time. Please reply when we will forward to you both answer, question and comment. Thank you. So we need to try now with the next speaker that we tried twice to connect is again Dimitrios Drakopoulos from Switzerland. I hope that now the connection will be fine and that we can hear him.
Can you hear me now?
Yes, yes, Dimitrios, we can hear you. Okay.
Okay, I will try to present without my camera on.
Okay, perfect. So at least we can see the presentation, so you can share the presentation.
Okay.
Yes, we have it presentation, so perfect. The audio is good, so go ahead.
Okay, so I'm sorry about that, but the connection of, I don't know, my connection was not good.
Okay, please, can you start again from the first slide?
Should I start from the first or I
Yeah,
can
would be
just
better because the presentation.
Okay.
Okay, good.
So, yeah, so I will present you about innovative cropping systems and biopesticides to prevent mycotoxins in wheat, and a brief introduction about the life cycle of Fusarium graminearum. So this is the most important mycotoxigenic species of Fusarium head blight disease complex. And here you can see a maize field after it was harvested. And here it's a very good environment for Fusarium graminearum to develop and to infect the subsequent wheat crop, which is wheat in this case. And we can see that the ascospores and the conidia infect the wheat heads. And of course, we are all aware of the yield and economic losses, as well as the mycotoxin contamination in the grain. So with the best available fungicide that are available now in the market, we can only reduce mycotoxin and disease by 40 to 50% on average in wheat. Therefore, it's very important to find solutions against that disease. So we have increased pressure of Fusarium head blight when we have maize wheat rotation. So farmers should try to use less susceptible hosts, but it's still a common practice. Also, when we have reduced or no tillage systems, we have an increased inoculum source, but they have quite a lot of advantages if we use reduced tillage systems. But how we can control residue-borne pathogens like Fusarium? So it's important to find solution in high-risk systems, and that was the aim of my PhD. I investigated different cropping systems and different plant-based biopesticides to control and reduce mycotoxins in wheat in a maize wheat rotation. So I had these three different approaches. I had three prevention strategies: use of cut and carry biofumigation, botanicals, maize intercropping, and cover cropping systems to suppress the inoculum of Fusarium graminearum and therefore to reduce the disease and mycotoxins in wheat. I also had a direct control strategy, but today I will talk to you only about the prevention strategies. I will go through one by one. So we had the cut and carry biofumigation and use of botanicals. And what we did here, we simulated a maize wheat rotation under no tillage. We collected maize.
Okay, we are not lucky with Dimitrios, so we need to move to the next speaker and to the next session. We have now the session of intervention strategies, and I would like to invite Giuseppe Meca from Spain to have his talk. He's a leader of work package 5 of MycoKey, mycotoxin intervention strategies. He will tell us about main outcomes from the MycoKey project. Please, Pepe. Giuseppe.
Hear me?
Yes.
Antonio?
Vai.
Antonio, yeah, just
Yes.
a moment. I'm trying to connect my camera.
Okay. Okay, share the screen and let's start.
Yes, I'm going to do that. Okay.
Mi dispiace che ti tirato in questa cosa, ma mi sembra...
Okay. Do you have a good audio? Could you hear me
Yes,
well, Antonio?
Everything is working.
Antonio?
Yes, we can hear you. Oh, mi hai tolto il coso.
Antonio?
Yes, Pepe, you are fine. We are fine. You are connected.
Yes, it's
I think that you have a good audio. I'm going to start my intervention. I'm going to explain the main outcomes inside work package 5 of the MycoKey project, in which from the half part of the project I was also the work package leader. Antonio, could you hear me?
Yes, Pepe. Pepe, don't stop. You are. We can see you. We can hear you perfectly.
Okay, so sorry. Okay, perfect. Okay. So as you know, the work package 5 was divided in 3 different tasks. We have from one hand the pre-harvest strategy based on the application of the biocontrol agent for the control of the Fusarium heat blight on field. The subtask of the production of the new generation fungicides that is in charge in general of the development of this subtask, and then the application of the electrolytes oxidizing water for the control of Aspergillus carbonaceus in grape and the control of the ochratoxin A production. The second big task of the work package 5 was based on the reduction of the fungal growth in silo system, and we have in this part the application of the ozone from the Stream Ozone company that's inside in our consortium, the application of some bioactive compound based on the reduction of the sporulation of Penicillium verrucosum and Aspergillus flavus growth in silo condition on maize, barley, and wheat, and we are in charge on this part of the project. And then the non-thermal plasma treatment and the application of the hydrogen peroxides also in cereals, and the application of supercritical CO2 and ultraviolet ray in order to reduce the growth of the fungal contamination main in cereals and also in another product like coffee and nuts. And then we have the 5.3 task based on the food biocontrol agent, development of the biocontrol agent for the cereal germination, and the integrated intervention process around all the development of the work package 5, and also the techno-economical performance. And finally, the last task was based on the food safety management and the HCCP associated to all the devices development and study around our work package. The first task that I'm going to present was the formulation of the biocontrol agent based on Chronostachys rosea, and on this task was in charge agroscope of Switzerland, led by the Susan Bogesgan, and this strain was isolated on Swiss agricultural soil, was already registered in the European Union. The growth was performed in 3-4 weeks, and this was studied that was available up to here at 5 degrees. The first step of the study was to understand the stability of the Chronostachys rosea against some degradation agents, like, for example, ultraviolet BOA, and after that, to investigate the effect of the biocontrol agent against the growth of Fusarium graminearum on maize in the experimental field of the agroscope. So what are the main outcomes of this part of the project? They discovered that the protection of the formulate with the sunflower oil reduced the degradation of the fungal preparation between 92 and 96%, and related to the result on the application of this strain on field, they observed an important reduction of the disease on wheat, and also an important reduction of the production of the mycotoxin deoxynivalenol up to 69% in comparison with the control. But they didn't observe any reduction statistically, significantly, from the statistical point of view of the mycotoxin zearalenone. And the second part of the application of the biocontrol agents was developed by the National University of Rio Cuarto in Argentina, led by Dr. Schulze. They tested several isolates from wheat and from soil, more than 350 strains. After that, with some in vitro assay, they studied and they performed more assay with 22 strains, and finally they characterized one strain or Bacillus valenciensis that was applied in lab condition in greenhouse, and after that was applied in field in several seasons from 2010 and to 2017, and this strain was applied on 2 different crops, the bread and durum wheat, in order to control the Fusarium with blight disease on this target crop. The application method was to spray the formulation over the weight spikes. The results are really interesting because we can observe in the both crop an important decrease on the severity of the disease and also on the reduction of the mycotoxin deoxynivalenol. In particular, on bread weight, we observed a reduction on the disease of the 48% and on durum weight of the 45%, and an important reduction of the DON of the 50% in durum wheat, and in bread wheat we didn't observe any production of this mycotoxin. I want also to remark that we are actually in contact with a company here in Spain in order to promote the exploitation of this strain, and we hope to obtain a formula that will be ready maybe for the real market for the application in field next year. Then, as Antonio remarked yesterday, the MycoKey project was also a big challenge and also a big opportunity to cooperate between the European Union and China. And inside the work package 5, there were some Chinese group that worked principally on the isolation of the anti-Fusarium graminearum microbes in order to develop isolate for the biocontrol of the Fusarium with blight disease in China that, like Antonio suggested in some market yesterday, it's a really big problem for the China market. And they started with the number of isolate of the 2,000, more than the 12,000 isolate, and finally the isolate that presented the antagonistic effect against Fusarium graminearum were close to 500 isolated, that corresponding more or less to 3.2% of the total isolate studied, and that are related to the microorganisms Bacillus, Pseudomonas, Panthera, Burkholderia, and the Streptomyces. And the first step of the research was focused on the identification of the inhibition zone of this isolate against Fusarium graminearum. And after that, they worked on 3 different steps. The first step were the antagonistic assay in plate in comparing the antagonistic effect of the pathogen against the biocontrol agent. They also tested the antimicrobial effect of the cell-free supernatant of the biocontrol agent against pathogen, comparing the result of the inhibition zone against the chemical fungicide. And after that, this strain were also tested in goat chamber on field against the Fusarium middlebride disease promoted by Fusarium graminearum. It's really, really interesting to submod that many strains present a biocontrol efficacy in field over than 50%, and some of these strains are really close to the market because they have a collaboration with some company, and maybe we hope that the same that I explained for Bacillus melenzensis in next year, maybe we will have this product ready on the market to be used on the Chinese field. And also in task 5.1, we have the application of the mustard-based botanicals for the control of Fusarium middlebride on feed. Why mustard-based botanical? Because, you know, mustard is a really interesting ingredient, and is a natural ingredient based on the mustard flour, and is really rich of several antimicrobial compounds. We have from one hand the glucosinolate, on the other hand the isothiocyanate, phenolic acid, organic acid, bioactive protein, and bioactive peptides. And also it's really interesting because it's a natural matrix. It's really cheap in comparison with the chemical fungicide. It's also environmental friendly, and also is safer for the operator in comparison with the manipulation of the chemical fungicide used on the field. And the first step of this research was in charge of this research was the Agroscope of Switzerland, and was carried out in cooperation with my lab in Spain, in Valencia. The first step was based on the characterization of the chemical compounds that produce the antimicrobial effects of the of several different mustard ingredients: the Tillecur, pure yellow mustard, pure oriental mustard, and oriental mustard brand. And we detected that the main compounds that participated to this phytocomplex of the antifungal compounds were principally the isothiocyanate, para-hydroxybenzyl isothiocyanate, and the allyl isothiocyanate, and several phenolic acid naturally present in the matrices. And then they tested also this ingredient for the control in vitro of the Fusarium graminearum, and we observed a really interesting decrease of the Fusarium graminearum growth in comparison with the control. And this result was also confirmed in the test due with pot experiment. We obtained also for some of these ingredients the reduction of the production of the mycotoxin, the oxynivalenol in grain, but we didn't observe any difference from the statistical point of view when we translate this experiment on field, maybe due to the instability of some antifungal compound present in this matrix, and there are more assays that are ongoing in order to have and to stabilize this compound and to propose to the market a real product based on this natural compound with a big efficacy against the Fusarium graminearum on field for the wheat treatment of the Fusarium in the blight. Related always with the task 5.1, we have also the application of the electrolyzed oxidizing water for the reduction of the growth of the Aspergillus carbonarius and the aflatoxin production in grape. This part, on this part of the research was in charge the Ispa CNR of Bari, in cooperation with the company that has the patent of the instrument in order to produce the electrolyzed water, that is the Nora SPA, localized in the south of Italy, in Apulia, in the region of Apulia. And this development was based in the production of the electrolyzed oxidizing water. This water was produced through the reaction between water and salt. We can use sodium chloride or potassium chloride, and using electricity, we finally obtain the antimicrobial compound potassium or sodium hypochlorite that is active again a broad spectrum of microorganisms. And in our work practice, we tested the efficacy of this compound against Aspergillus carbonarius. Giancarlo performed a test on in vitro against the conidia germination of the Aspergillus carbonarius using different concentrations of the compound and also comparing the result with the chemical fungicide switch, and also was the result obtained in vitro were confirming on plate using weight contaminated with Aspergillus carbonarius. And finally, we demonstrated also the activity of the electrolyzed oxidizing water in field on Primitivo wine grape, typical of the south of Italy, using several concentrations of the electrolyzed oxidizing water and also the antimicrobial compound switch. If we focus our attention on the test carried out in lab, we observed an important reduction of the fungal growth of the Aspergillus carbonarius with an increase of the electrolyzed water usage. This result were also confirmed by the test carried out on grape in plate, because we can observe an important reduction of the infection of the grape by Aspergillus carbonarius that is proportional to the concentration of the electrolyzed water used. And finally, these results were also confirmed with test on field, as I said before, in Primitivo field, where we observed also an important reduction of the production of the ochratoxin A. And one of the main achievements of the work package 5 inside the task for 5.1 was the development of the new generation compound, new generation fungicide developed by Syngenta. These new generation compounds were tested in two fields, in Bologna field and Torino field, on 5 varieties of wheat, using 3 different treatment check standard and Syngenta compound with 4 replication in 120 plots. I'm just going to do a brief vision, because then we will have a presentation by Syngenta on that. They observed an important reduction of the severity of the Fusarium heat blight on the ears, considering the main of the 2 locations in 2019, comparing the Syngenta compound in comparison with the control. And also they observed also an important reduction of the ppb of the deoxynivalenol produced by the Fusarium graminearum, considering all the 5 varieties tested in the field of Torino. And then we are going to move with the second part of the presentation, was a focus on the application of sun treatment in order to reduce the growth of the fungi producer on the mycotoxin during the storage of the cereals. And the first approach was is based on the application of the ozone. On this part of the project was in charge the company Stream Ozone of the Ukraine, and was in charge on this part of the project Dr. Sergi Pugac. And the ozone, you know, is a really important compound that is used from many years ago for the control of several kinds of the microorganisms during the storage of several food products. They have a patent on the application of the system for the application of the ozone in silo system, and they worked basically on the control of the Aspergillus flavus and Penicillium verrucosum on maize and on wheat, and on a volume of a small silo of 70 kilograms. I just going to do a brief introduction on this part of the project, because Serge is just the next speaker of my presentation. What they observed was in corn an important reduction of the infection of the Aspergillus flavus with a big reduction of the growth, as we can observe in this slide, and this reduction was also observed for the Penicillium verrucosum, also on wheat, demonstrating the efficacies of the application of the ozone to reduce the growth of these two mycotoxigenic fungi on these 2 measures that are object of our work package. And finally, we are going to present also some details on the research that I carry out in my lab in the University of Valencia we worked in work package 5 in order to develop a new antifungal device in order to reduce the growth of the Aspergillus flavus on maize and Penicillium verrucosum on barley, based in the bioactive compound allyl isothiocyanate. And this device was validated using a small silo in lab condition, in small silo condition, and also in a real silo condition. Just a brief introduction on the device. The device is composed by a bioactive gel, that is hydroxyethyl cellulose, that is introduced in a small petri dish with the antifungal compound that is the allyl isothiocyanate. Allyl isothiocyanate is a really interesting compound. It's a gaseous antifungal, natural antifungal compound. It's the main antifungal compound produced from the natural matrix that is the oriental mustard flower, and is produced through the conversion of the glucosinolate simigrine. That's one of the main glucosinolate present in this matrix. And it's really interesting because it's active again a broader spectrum of microorganisms, is active again fungi, but is also active again several kind of other microorganisms like bacteria and yeast. And we introduced inside the jar our device, and we treated 300 grams of corn, barley, and wheat inoculated with Penicillium verrucosum and Aspergillus flavus. And after that, we closed the jar, and the allyl isothiocyanate is really volatile and occupied the space of the jar, reducing the growth of the fungi and also the production of the mycotoxin. We, in the lid of the jar, we put also a setum in order to control with GCMS, with GCFID, the volatilization of the gas, and also we put it in air escape for the respiration of the cereals. What about the first part of the result in this small in this lab condition? We obtained that after the 30 days of the treatment, because we incubated the cereal with the fungal contaminants during 30 days, a concentration of the allyl isothiocyanate in the heat space close to 0.25%. And it's really important to submark that this concentration is really close to the minimal inhibitory concentration of the ATC. Looking for the microbiological analysis and the reduction of the fungal growth around the incubation, we observed an important log reduction for corn, close to 4 log, and we didn't observe any growth of the Penicillium verrucosum on wheat and on barley at 30 days of incubation. But also we observed a big and an interesting reduction of the production of mycotoxin for aflatoxin B1 and for aflatoxin A. We moved it for aflatoxin B1 from 8 ppb of the control until 0.1 ppb, and for aflatoxin A from 0.3 ppb until 0.1 ppb. And after that, we validated the system in a small silo condition. We moved it from the a small jar, to a plastic drum where we tested 70 kilograms of corn or wheat and also barley, contaminated the corn with Aspergillus flavus and the wheat and the barley with the Penicillium verrucosum. And the cereal matrix was present in the upper part of the drum, where in the lower part we introduced our device. We moved it from a small petri dish to a bioactive Tupperware with the same proportion of the gelling agent, that is hydroxyethyl cellulose, and the antimicrobial compound allyl isothiocyanate. Also, the drum was connected to an air pump in order to facilitate the evaporation of the allyl isothiocyanate and the contact of this antifungal gas with the contaminated matrix. What about the result? We still obtained an important reduction of the fungal growth. In this case, we observed the fungal growth during 60 days. We observed the reduction of the 2 log of the growth of the Aspergillus flavus on corn, and also a reduction of more or less on one log for the growth of the Penicillium verrucosum in wheat and in barley. And we also observed an important reduction of the mycotoxin, the ochratoxin A, and wheat and in barley, in particular, the control of the wheat presented more than 300 ppb of the ochratoxin A, and we observed a reduction until 72 ppb. And for the barley, we moved from 200 ppb of the control until the 15 ppb of the barley. So we observed also an important reduction of this mycotoxin. And finally, we demonstrated also the efficacy of our device in a big silo condition. This part of the experiment were carried out in cooperation with another company present in work package 5, that is the company Burmalt, and we developed this part of the project in the facilities of the Burmalt in Ireland, and we moved from 70 kilograms of the barley to 50 tons of barley in silo condition, and also we switch from the bioactive Tupperware to a more technological device based on a bioactive can, where we introduced always the gelling agent with the same relation of the quantity gelling agent and the antifungal compound allyl isothiocyanate. And we put this device in the main entrance of the fan, and we connected the fan to the silo in order to promote with the air the volatilization of the antifungal compound allyl isothiocyanate, and to put this antifungal agent in contact with the contaminated matrix. What about that? The result related to this part. As I commented before, we observed an important reduction in lab condition. We still observed an important reduction in small scale condition, and in big condition, in big silo condition, we still observed a reduction of the fungal growth. In fact, after 4 weeks of the incubation, of 4 weeks of treatment, we still observed a reduction of the 0.2 logs of the Penicillium verrucosum on barley. It's really interesting to submark. This is still important to obtain. It's important to submark that this quantity of the log, it's important because corresponding in the real market to a reduction also of the mycotoxin produced by this fungi. I want also to submark that this device is patent. We patented last year this device here in Spain, and now we are going to perform some more experiment, also always in cooperation with Burmalt, in order to test with silo or bigger condition in comparison with the silos used in this study. Summarizing the main outcomes obtained in work package 5, we can conclude that the clonostatic rosea reduce significantly the growth of the Fusarium graminearum in vitro, and also was important to reduce the production of the mycotoxin, deoxynivalenol, and the Fusarium wilt-like disease on wheat. Bacillus valenciensis was also another important biological control agent developed by the National University of Rio Cuarto in Argentina, demonstrated a good activity against Fusarium blight on durum and bread wheat, and we observed an important reduction of the disease, and also the important reduction of the production of the mycotoxin, deoxynivalenol, and we have also an important TRL for this biocontrol agent because you are really close to introduce this product on the market. And then some isolate of anti-Fusarium microbes isolated by the Chinese partner reduce the Fusarium blight incidence on wheat more than 50%. We obtained an interesting result on the application of the mustard powder in vitro and also on pot on the control of the Fusarium graminearum growth and also the production of the mycotoxin deoxynivalenol, but we have need or more data related on the application on field and on the stabilization of this preparation in order to preserve the ingredients on the degradation of the bioactive compound. We obtained a really interesting and with really high TRL result on the application of the electrolyzed oxidizing water for the reduction of the Aspergillus carbonaceous growth and the ochratoxin A production in wheat. Syngenta developed a new generation fungicide that reduced the severity of the fusarium mid blight, and also the production of the mycotoxin deoxynivalenol in grains, the application of ozone in pilot scale significantly reduced the contamination of Aspergillus flavus on corn, and also the Penicillium verrucosum growth on barley. And in our lab with our device, we demonstrated that our device based on bioactive gel containing the fungal, the antifungal compound allyl isothiocyanate reduce the growth of the Aspergillus flavus and Penicillium verrucosum in lab condition, obtaining a really big log reduction of these two fungi and also a reduction of the mycotoxin produced. We observed also an important reduction of the growth of these two fungi in small simulated silo conditions that were confirmed also in real silo condition. I want to conclude my presentation, say thank you to all the partners, all the main partners involved in the work package 5, and also to the Ispa CNR to give us the opportunity to participate in this big project, this big challenge that for us was the project MycoKey. And also I want to say thank you to Antonio Logriego, Antonio Moretti, and all the steering committee to give me the opportunity at the half of the project to be the work package leader of this really important work package after the Dottora Laitila leave our work package. And also, thank you very much to all of you for your attention, and I'm completely available for questions.
Thank you very much, Giuseppe. And now we have just a quick question.
Yes, we have a question from Francesca Fanelli. Is the hydroxyethyl cellulose authorized as food or feed additive? Does it have any effect on cereals quality or nutritional properties?
No, the hydroxyethyl cellulose that we use is approved for several uses. We use an ingredient approved for food, for cosmetic, for medical approach, for everything, basically. And we also tested the influence of the compound against the several biochemical parameters, including the chemical composition, nutritional composition of the cereal, and we didn't observe any change. Also considering the flavor different that could be influence this application.
Thank you very much, Pepe. And now, thank you very much for his answer and presentation. And now we go further and we give the words to Sergei Pugak from Ukraine. His talk will be about mycotoxin intervention strategies inhibiting effect of ozone.
Good afternoon, Antonio. Can you see me?
Yes, we can see you.
Okay, and here I see. Okay, good afternoon, everyone. It's a pleasure for us to present the result of our activity in the MycoKey project, and it devoted the inhibiting effect of ozone for fungal and mycotoxin decontamination. And the presentation consists of brief information about streamozone, about our main tasks in the project, about our research results, and testing of lab scale, pilot scale, and industrial scale system, and conclusions of our activity in the project. First of all, we are a spin-off company originally from National Science Center, Kharkiv Institute of Physics and Technology. It is the largest nuclear research center in Ukraine and the largest institute in the physics science. And first of all, we are physicists, but we are interested in different applications and agricultural applications. One of the most interesting for us and for our technologies, for our ozone technologies. And so we are a small R&D and manufacturing company that provides ozone equipment, ozone systems for different applications, for air treatment, for water treatment, and for agriculture. We were involved in work package 5, namely task 5.2, post-harvest strategies during storage, and our main task were manufacturing of lab-scale ozone prototype and laboratory treatments of wheat and barley contaminated by Aspergillus flowers and Penicillium nordicum and verrucosum. As well, we have trials with maize. After that, manufacturing a pilot ozone system and trials of ozone efficiency in inhibiting of toxigenic fungi during storage and mycotoxins in wheat and barley. And finally, pilot ozone system and its testing in industrial equipment, industrial environment, sorry. Our first step was laboratory ozone treatment and decontamination of fungi pure culture. We used Aspergillus flowers and Penicillium nordicum pure culture, and we found that both of these strains killed by ozone and reincubation of treated fungi doesn't lead to fungal growth. And this stage allowed us to find preliminary modes of treatments that we use in further treatments on grain. The next step was laboratory treatments of wheat and barley grain. We use small batches like half of 1 kilogram. Wheat contaminated by Aspergillus flowers and barley contaminated by Penicillium nordicum. We used treatment during 2, 4, and 6 days, and after that we put these samples to thermostat and observe contamination level in 3 and 7 days after treatment. So we found that the number contaminated grain of wheat was decreased from 100% to 38 in average, and barley was contamination was decreased from 64 to 16%. After that, we move for larger treatments. We used silo simulate approximately 100 kilograms of grain, and we use maize and wheat. Maize was contaminated by Aspergillus flowers and wheat by Penicillium verrucosum. And we used the same treatment modes as we used earlier, and was found that ozone inhibited the growth of fungal contamination and the number of colonies in 1 gram of grains in average by 3 times, or 60–70%. We mostly assay the influence of ozone on fungi growth, but we made experiments with mycotoxin decontamination. We used maize that was highly contaminated by Aspergillus flowers, and looked for aflatoxin B content in control samples and samples that were treated by ozone during 3, 4, and 5 days, and found that mycotoxin, aflatoxin B decontamination, it correlates with the fungi decontamination. And the result was the content of aflatoxin B decreased by 60%. That is very close to fungi decontamination, Aspergillus flowers decontamination. Okay, and the next step was larger silo simulator. It was 1,000 kilogram of grain, and we use wheat that was contaminated by Aspergillus flavus. It was trials during 6 months, started in 2019 and at the end of middle of 2020. And we used ozone treated mode, like 3 days of ozone treatment twice per month during this period of time. And the average effect of growth inhibition for Aspergillus flavus was 75%. And the next step should be the industrial trials, and we had some difficulties to arrange these trials with wheat, so we had very huge silo from our partner. It was they have like 6,000 tons as a minimum silo, so it's too large for our equipment that we can provide for this trial. So we found another decision. We arranged this industrial trials validation in Ukraine with sunflower grain, and we used 800 tons of sunflower. We manufactured specific systems that consist of ozone generating module and ozone injection system, and used the same modes as for wheat and barley that we used previously during 5 months when this sunflower storage facility. This sunflower was contaminated by Fusarium graminearum and Aspergillus flavus, and at the end of storage and process, we shown that the average inhibition of fungi growth during this 5 months was 50%. That is close to our previous result on wheat and barley, as well as maize. So finally, our conclusions, we shown lab scale ozone treatments as well as pilot scale trials that ozone inhibiting inhibition of Aspergillus flavus, Penicillium norbicum, and Penicillium verrucosum by 60 or 70%. Lab scale ozone treatment shows that inhibition of aflatoxin B level is 60%, and this level is correlates with inhibition of fungi strains. We identified the treatment mode that allows us to get these decontamination results, and this mode is ozone concentration on the level of 100 milligrams per cubic meter, and exposure time is not less than 72 hours. And our conclusion that ozone decontamination is an option for fungi growth control, and it doesn't lead to full inhibition of fungi and mycotoxin. And finally, we validated our system on the industrial scale and shown that the results are very similar on the biggest batches, like 800,000 tons. Okay. And finally, I would like to thank from our Streamazone team to all MycoKey team, work package leaders, Antonia, first of all, and of course Pepe and Aria and all of coordinators, members for the fruitful action and promising results that we achieved in this project. Thank you very much.
Thank you very much, Sergi. And there is a question.
We have a question from Sara De Sager. Do you know if aflatoxin degradation products are formed?
Actually, no. I know this product. I don't know this product. Yeah.
Ah, okay. Thank you. I have a quick question also. Do you have did you know if there is any influence of the food matrix, let's say, in the efficiency of the method? For example, towards sunflower, maize, wheat, the ozone works in different way, or there is no influence of the food matrix?
Actually, we didn't investigate food matrix. If you mean changes of
the commodity.
Sorry?
Of the commodity, of the food that you treat.
Okay. Actually, we used our treatment for different kind of food during storage and process. It's vegetables or some grains, and what we found that we have like very close or similar results on fungi decontamination, but we in our work we didn't investigate internal quality of food, so changes.
Okay, thank you very much. Thank you very much, Sergio. Okay, we move to the next speaker. The next speaker is Levent Yasin Kurt from Turkey, and his talk is about mycotoxigenic fungi mitigation through cold plasma treatment.
Hello.
Hello, we can see you, and
you see me now?
yeah, you can share the presentation.
Mm-hmm. Okay.
You can.
I think it came now.
Yeah, we can. If you put it in modality presentation, we will have a full screen of presentation.
Mm-hmm. Okay. Hello, I am Levent Yasin Kurt. I am from TÜBİTAK MRC, Turkey. And my presentation's topic is mycotoxigenic fungi mitigation through cold plasma treatment. In this presentation, first I will start with definition of the problem, introduction to plasma systems, and I will give our pre-study results, and then I will give our latest study results, and I will also share with our conclusions and future needs. So, you know, every study comes from a problem. Firstly, we also shall mention our problem, which you probably already know. Our problem is 1/4 of crops produced in worldwide are contaminated with mycotoxins. And we know there are more than 500 mycotoxins are identified, which causes mass losses, mass food losses, and expensive healthcare problems, and even death in some times. Traditionally, for solution of this problem, decontamination processes are used, and most of the decontamination processes used in this problem comes with heat treatments. It's a heat treatment, but heat treatment process itself causes a lot of heat-sensitive components, like vitamins, volatile nucleic components, etc. And it can cause undesired changes in the organoleptic properties of foodstuff. And therefore, here comes a new objective for our researchers, which is non-thermal, non-destructive decontamination methods. And in our study, we used cold atmospheric plasma systems. Okay. When we talk about plasma, as you may already know, again, plasma is the fourth state of the matter, and it's a relatively new invented subject. It's basically a partially or fully ionized gas. And I am giving these definitions because when we talk about cold plasma, the term cold actually is not the conventional means of cold. I can say it from here. When we talk about fully ionized plasma, we are talking about solar explosions, laser fusion plasma, and we are talking about 10 to 6 to 10 to 8 centigrade degrees temperature. And also, when we talk about partially ionized plasma, especially quasi-equilibrium plasmas, we are talking about 10 to 4 centigrade degrees temperature, which can be sampled like arc plasmas or plasma torch, and you can see from the pictures, it can actually be used to cut through metals. And when we talk about cold plasma, actually we are talking about a temperature between 30 degrees to 700 degrees. And I know this is not actually cold, but it's actually colder than the other plasmas. And typically we can show samples of cold plasma like corona discharge, the electric barrier discharge systems, and atmospheric pressure plasma jet systems. The usage of plasma for decontamination results from three formation coming from the plasma generation. First one is UV radiation, which is causing a DNA damaging. The second one is heat damage, also caused by, especially in the long duration applications. And the third one is reactive oxygen species and other free radicals formed during the plasma generation. And especially in the literature, it is mentioned that these high energy generated low life reactive components are the main actors in short time decontaminations. And again, you might already know free radicals are highly reactive molecules that have a strong oxidative effect and causes lipid oxidation. And through this, they primarily damages the cell membranes and cell walls. And then after that effect, they can also damage the DNA. And again, literature says us that gram-negative bacteria are more vulnerable than gram-positive bacteria, and prokaryotes are more vulnerable than eukaryotes. This is mainly because of the DNA protection systems and lipid content in cell membranes and in cell walls. And our target was Aspergillus flavus in our study, which is already a very hard to deal microorganism. We used a plasma system produced by a plasma trade company from Germany, and our system consists of speed control units, plasma generator, plasma nozzle, plasma rotary jet, conveyor belt with corona system, and exhaust blower for corona. And in our system, actually, we can adjust the voltage, frequency, plasma cycle time, and air pressure. And in pre-studies, we started with corona system trials, and we used a design expert program with central composite design model. We prepared 20 artificially inoculated wheat samples with their blanks, with their parallels. And in the corona system, we also know that the distance between the electrodes are very important. Therefore, we also know that for forming corona, we need a 3 to 10 millimeter distance between the electrodes. Therefore, we used both 3 millimeters and 10 millimeters distance in the trials. And this was the results. As you can see from the pictures, almost all our samples was burned after 10 seconds. Our electrodes becomes unusable, and the most surprising one was not that. Most surprising result was, even though they were burned, the reduction rate of fungi was no more than 0.4 logs. And this makes us think if the plasma are suitable to use in Aspergillus flavus or not, then we need to check the plasma efficiency of plasma. Therefore, we directly applied rotareject plasma to petri dishes, and after 3 days and 7 days of incubation, again you can see from the pictures that system was actually effective. But the affected area was very limited with the contact point of corona flame, plasma flame. Therefore, we need to return to the theory. And actually, we needed to build a new hypothesis for this trial. And we knew that free radicals were the main actor in decontamination. So we plan to increase the free radical content of plasma atmosphere. We also know that hydroxyl radicals were more effective than the others from literature. Almost in every paper it's mentioned that. So we decided to use water vapor to increase hydroxyl radicals in the system. But for combining water vapor, plasma, and samples needed an extra equipment. So we designed a new chamber for it. It was a very simple chamber, actually. In this chamber, plasma flame was met with water vapor at the top of the chamber, and plasma-activated water vapor is generated in the chamber. And we are feeding the samples from the top of the chamber, and during the free fall of the samples, all wheat grains are in contact with plasma-activated water vapor. And after the end of the chamber, we waited with sterile cups, collect all samples, and this one cycle, this is named as one cycle. And to see the effect, we use multiple cycles with the same samples. And for better comparison, samples were both treated in dry air plasma and moisturized air plasma systems. And when you look at the dry air plasma system, you can see that our results were not good either. And they were very similar to corona system trials, which which I mentioned in the previous slides. But when we look at the moisturized plasma system, at the 10th cycle, we saw a 2-log decrease in the Aspergillus flavus. It was a very significant decrease for us. But because of the using of moisturized air, we also check the moisture content and the temperature of the media too. Temperatures, I need to mention that the temperatures were measured immediately after the treatment with no waiting, and even it increased to 70 degrees after the 10th treatment. After a short time, it decreased to room temperature, and the moisture content, as you can see from the results, were not increased. So After these trials, we concluded that the atmospheric pressure plasma jet activated the water vapor and formed plasma activated water vapor. And plasma activated water vapor contains enough hydroxyl free radicals to reach wheat grains, easily in the design chamber. And the contamination ability of the system has been increased without causing a significant increase in moisture content of the grains. And the temperature of wheat grains were not increased enough to give them sensory problems like burnt odor, color, and etc. And after this study, we saw that we also concluded that the effect of different chamber designs to the contamination capacity of system shall also be investigated. Process optimization studies shall also be conducted. And the effect on large-scale production shall also be investigated. And effect on other mycotoxigenic nodes shall also be investigated. And thank you for your attention. That was my presentation. Thank you for everything. Thanks again for your attention.
Thank you very much. Thank you very much. And there is a quick question because we are running out of the time.
Yes, the Tiger asks, is there any effect on mycotoxins?
Yeah, in our trials, we also looked the mycotoxins, but the results were not good to mention. It was very difficult to obtain results because we couldn't find naturally contaminated samples. Therefore, results were nothing. There was nothing to present in here.
Okay.
After
thank you very much. And now we go with the last talk. That is Dr. Yun Chen from China. His talk will be integrating fungicides and biocontrol agents to manage Fusarium head blight of wheat in China. And is one of the example of the great interaction between European and Chinese groups on this along this project. So Yun, when you are ready, you can start with your presentation. And
can you hear me?
Yes, we can hear you.
So how to share my
screen is the screen is shared. You need only to put top. You can start.
Okay. Hello everyone. So my name is Su Ying from the Zhejiang University, China. So it's great to honor to give a short talk here. So our lab mainly works on the Fusarium head blight. So today I will be give a short talk about the. Okay, the short talk about the integrated fungicide biocontrol agents to manage FHB wheat or China. So I will briefly introduce the current situation of the Fusarium head blight in China, and then tell you how we use the fungicide to control the Fusarium head blight, and also show our recently progress on the biocontrol FHB. And at the end of my talk, I will be give a short summary and a prospect on the integrated strategy for the FHB in China. So I think that all you know Fusarium head blight caused by several Fusarium species, especially in China. The Fusarium graminia and Fusarium asiaticum is the major pathogen of the Fusarium head blight. I get this slide from the Dr. Hao Zhang. So if you see the map in China, So you found the south of China, the FHB are mainly caused by the Asian twosome, but the north of China is mainly caused by the Gramminea. So I think so it's maybe caused climate and the host. So the FHB is now a bigger problem in China. So the yearly occurrence of the FHB in China is more than 5 million hectares. That means it's 20% of the total planted area of the wheat are affected by the FHB. So as you know, the FHB not only cause the ear loss, but also the mycotoxin. I just give for example here. So in 2010 and 2012, we did a survey in Jiangsu province. We find that DOM positive the wheat sample is more than 70%, and the maximal DOM concentration is 41. That's very dangerous for us, right? So we think that the FHB epidemic is more frankly caused by some major factors. Here is the summary about the thing. First, we don't have the resistance wheat cultivars. During the last 5 years, we tested more than 1,000 wheat cultivars in China and found more than 90% of the wheat cultivars are susceptible to the Fusarium hate blight. And compares with the previous years, we found that there is the more rain and high temperature in the May. You know, in May in China, the wheat is flower stage, so that's caused some more severe. So we also find that the store returning provides the more nutrients for the Fusarium species, the sexual and sexual development. That means they can provide the more abundance, primary inoculum. Also, series-based rotation, the weight and maize rotation and the rice wheat rotation also provides a more environmental conditions for the Fusarium. So we think, and let's back to the triangle. We don't most of the wheat are susceptible to the Fusarium hate blight, and there is enough inoculum and suitable environment. That's why FHB epidemics are more frequent in China now. So consider the FHB cause food safety and food security. Now China has listed FHB as one of the national first-class disease last month. So that's the first time China have this list. So we now I tell you how we use the fungicides to control the FHB in China. So we have generally we have the 3 kinds of fungicides for the FHB control. Well, first is Captazam. I know it's already banned in Europe, but still widely used in China. The second one is Phenylmecryl. It's only commercialized in China and so very good efficiency to FHB. The last one is Azum categories fungicides. So Captazam have been used more than 20 years for the FHB management in China. It's still used now because it's more efficiency. It's efficiency in some area in China, but you know the Captazam resistant field population is already increased. Our labs connect a wide isolate from the field and test the sensitivity to the Captazam every year, and more than 6,000 strains test per year. You find that the resistance population is rapidly growing, right? So especially in the Jiangsu and Anhui province. So we, of course, if we the best ways to don't use the capital, but you know, the capital is very cheap. The farmer is very welcome to use the capital. So we developed a strategy to for the capital resistance management. We did a very simple experiment, just mix the resistance strain and the sensitivity strain with different ratio. Then you spray the capital to see the efficiency. So you see if the resistance population is more than 10, so that the control efficiency is decreased very rapidly. So at the end, we get a guideline for the farmer. So don't use the capital only when the capital resistance is more than 5%, and stop use the capital when the resistant population is more than 10. Okay. Let's see the second fungicide, the phenylmecro, is China only. It's developed and by the China and only commercialized in China now. We try to register it in the European and United States. So phenylmecro is Fusarium species specific fungicide. It's only kills Fusarium species, not other test fungicide fungi, and it also shows a very good the mycelium growth inhibition. It's not only control the FHB disease, but also reduce the mycotoxin. So a few years ago, we find the target of the phenylmecro as the target of the phenylmecro is MyCO1. Is MyCO1. Phenylmecro can directly inhibit the ATP-based activity of the MyCO1. And also we already know why the phenylmecro can inhibit the dorine production. As you know, so dorine uses acetyl-CoA as precursor and many try enzyme to synthesize the dorine. So, but nobody knows where is the cellular place for the dorine biosynthesis. We use TRY1 as a marker, labeled with the GFP to see where is the localization of the TRY1, and we found the TRY1 during the inducing mycotoxin inducing media. TRY1 GFP mainly localized in this and its organelle. So finally, with the phenylmecro fungicides can totally inhibit the formation of this this organelle. We call this dorine toxin. So finally, we find the toxin is kind of remodeled ER. So we draw some model during the dorine biosynthesis, the ER remodeling. So so the MyCO1 directly interacts with the actin, provides the energy for the ER remodeling. Now phenylmecro fungicides can directly inhibit the activity of the MyCO1, then stops the biosynthesis of the dorine. So for the ozone, so we also check the resistance population in the field, but now we don't until now we don't see the high level of the resistance isolated from the field. But one thing that I want to point here is we find no dosage of the ozone category fungicides will stimulate the dorine toxin formation and stimulate the dorine production. So we give a suggestion for the farmers, you should use the high dosage of the ozone category fungicide for the FHB management. So the next question is, what's the exact best time for the fungicide application? We also did a very simple experiment, spray the fungicide at different growth stage. As you see, the early and the middle flower stage is the best time for the fungicide spray. So give a short summary for the chemical control FHB in China. So we have the 3 category fungicides can be used for the FHB, but also use phenolmacro and some azure, also some mixture of them. And application time is the early and middle stage, flower stage. The frequency is 1 or 2. That depends on the weather. So also point here, when you use the unmanned air vehicle for spraying, the formulation of the fungicide should be modified, and the dosage should be increased. So after the chemical control, we also use research on the FHB biocontrol. So we isolate the biocontrol strain from the wheat-associated environmental. And until now, we have already screened more than 15,000 strain, and combined with the growth inhibition and targeting of the DOIN toxin formation and the field experiment, we have the 7 very good potential biocontrol agents. They are Bacillus, Pseudomonas, Pentoya, and Prochlorhodere and Streptomycin. So we also identified some activity compounds from this biocontrol strain and to the molecular mechanism, how this bioactivity compounds can kill the fungi and or inhibit the DOIN production. So here is just to show us example. The ZG16 is Pseudomonas strain. Pseudomonas chronography is showed very good biocontrol efficiency in vivo and in vitro experiments. So this biocontrol strain secreted the phenazine compounds and is targeting the fungi histone acetyltransferase G5 directly inhibits the activity of the acetyltransferase. So because the histone acetyltransferase is the main component of the Sake complex, is histone acetyltransferase, right? So because Sake complex is essential for the fungi infection and DOIN toxin formation with DOIN production. Now the biocontrol agents ZG16 can screen the phenazine compounds and get into the fungal cell targeting the G5 and totally inhibit the activity of the histone acetylation. Then inhibits the gene transcription of the DOIN and DOIN biosynthesis gene tri. So that's biocontrol mechanism of this strain. So we also try to mixture the chemical fungicides and biocontrol agents. Here is I just show you one example. This is the biocontrol agents. The Pseudomonas can grow in the plate on the plate, supplemented with different fungicides. So it's compatible. So we also mixture them with the fungicide and sprayed in the field for the FHB control. As you see, a mixture with the half dosage of the azure category fungicides with the biocontrol agents, you will get the very good control efficiency. So we can reduce the dosage of the chemical fungicides. So also now in China, we have the Bacillus already registered for the FHB control, and also the chemical fungicides with biocontrol agents mixture already registered for the FHB management. So as said in my talk, I'll just give a short perspective on the integrated strategy for FHB management. As you know, the resistant wheat cultivars is the best way to control the FHB. As you know, in China, the Agenda Agriculture Group already finds a new resistant gene FHB7. So it maybe can be used for breeding, plant breeding. Our lab also do some genetics with cultivars. Here is we use the host to reduce the gene silencing to created resistance with cultivar. You see this transgenetics weight shows very reduced 15% of the disease and also reduce the DOIM production. So also developed some new fungicide here. So our lab already finds new chemicals compounds can be used. Chemical fungicides is also targeting MyCo1. It's a new one. It's not totally different with phenylmacrol. We also use screening more biocontrol agents and then try to reduce the fungicides in China. We think we can combine with the biocontrol agents and chemical fungicides to management the FHB because biocontrol can reduce the primary inoculum, and the fungicides can be sprayed during the flower stage. So we can use this integrated approach to control FHB and reduce the DOIM production. Okay, that's my talk. I want to give thanks to my collaboration, Kobe Kissler, and also other students in lab, also MycoKey partner, and some funding in China. So thanks for your attention. This is my talk.
Thank you very much, Yoon. Very interesting talk. I have just a quick question to ask you about this morning. Susan Vogelsang was in a comment to a question, say that to integrate all tools we have, biocontrol agents, fungicides, resistant varieties, and eventually crop rotation. So the integration of these tools is the key factor for better fight against the fusarium and blight. Would you make a comment on this?
Okay, so we did a lot of experiments to try to figure out what's the exact times we should use the biocontrol agents. So we think if we spray the biocontrol agents at the with flower stage, that is not good because the disease infection is very rapid, right? So we think we can use the biocontrol agents to spray on the plant straw to reduce the primary inoculate. So spray the biocontrol agents in the wheat field. So after harvest, then use the chemical fungicides for the flower stage spray. And also we think we mixture the biocontrol agents with fungicides sprayed at the flower stage could be more efficiency because the bacteria can produce many polysaccharide and something very stick. It can help the fungicides on the wheat head. So we think that's what we think.
Okay, thank you very much for presentation and answers. And we have a further question. Sorry, from the audience. We can just close with this question.
Stefania Somma and Mario Macedo, they ask, did you evaluate the survival of BCA like bacillus on crop residues?
Sorry, I can hear very clearly.
Okay, I will try again. Did you evaluate the survival of BCA like bacillus on crop residues?
I try to repeat. Did you understand?
Would you mind repeating?
Okay, the question is, did you evaluate survival of the biocontrol agents of the crop residues, the efficiency in their survival?
Okay, so actually we don't check the survivals until now. So I think for the bacillus species biocontrol is as good, because I already do a lot of biocontrol disease use the bacillus. It's very good survival in the environment. For the Pseudomonas or some gram-negative strain, it's not good. It will be sprayed on the plant residues. It degrades very sharp. So until now, Bacillus species is the best choice we should try, because it's a very good biocontrol efficiency and also survives in the plant's residue with because they can form spore. And for the gram-negative, we still try to figure out the best formulation could be improved as well.
Okay, thank you very much again. So we can stop this session, and we have now our digital break because we are a bit over, just 15 minutes, but mostly because we need to make some technical connection with the speakers that will follow in the voices from food and feed industry session. We would like to start again the conference at 13:45. So please come back at 13:45, and we will have the session of voices from food and feed industries. And then we have the closing of the day with the talk of Jean Loudorne from EFSA, which will talk about an important point, risk assessment of combined exposures to multiple chemicals. So, see you later.
Thank you so much, Antonio, and thanks for the invitation to you and all the organizers. It's a very big pleasure to have the opportunity to present to you. AflaZero is a relatively new startup. We've been incorporated in 2017 here in Kenya, as you were saying, and we basically were aiming at removing aflatoxin from grain and pulses. The technology has been developed by our technical director as well as our inventor as part of the co-founder of AflaZero, which is also Italian, which is Franco Mazzariello, and who has been working for more than 10 years in research and development in Italy to develop a solution to aflatoxin, partially also funded by the European Union. But the particular important thing is that he was trying to respond to an emergency coming back from the time of the 29 during the food crisis, the global food crisis, because the hazelnut, which were imported at the time from Turkey and other countries, were coming with a higher level of toxin compared to the one allowed at the time in Italy. So after 10 years of testing and review, we have now decided to move to an industrial application. We've been testing extensively in the last 3 years with a high level of toxin, the plants, which I will show in a very short time. And we've seen arriving results which are pretty impressive. The plant is an ozone-based post-harvest solution. We are arrived to eliminate quite consistently up to 98% of the toxin present in particular maize. Most of our research has been done in maize because we have tried to solve a problem which in Kenya has been massively high in terms of level of contamination of maize. I will tell you more. The interesting thing that we have done a number of research during the last couple of years, we didn't find any toxic residues, both as a byproduct of the toxin as well as byproduct from the maize treated. The situation in Kenya, as I was mentioning, is effectively extremely high. We've seen, I mean, in a previous life, I was the country representative here for the Food and Agriculture Organization, and we did 3 years of research project on aflatoxin, and we've seen up to 80% of the products in supermarket highly contaminated by aflatoxin. The numbers are always very variable. Unfortunately, in a number of African countries, the number are not that trustable. But the reality is that we've seen a massive amount of toxin. And in fact, our major investment in the last 4 years of research and development was to raise the capacity of our plant from few tens of ppb to hundreds of ppb in terms of material to be treated. The good news for us in Kenya is that Kenya is a very serious country in terms of aflatoxin management. So is one of the very few that is actually burning the material which is which happen to be contaminated. Normally they seize it and they incinerate above 800 centigrade in a proper way. So we propose to the government of Kenya, instead of incinerate and destroy and create further CO2, to give us that product, allow us to treat it, and initially to use it just for livestock feed, although we have been pretty reassured by the fact that the quality of the grain that were coming out from our plant were much higher than the quality of the maize that was used for human consumption, and with a very, very low amount of toxin and zero amount of fungi on the treated grain. In that sense, we have been also starting discussing with commercial companies to solve the problem that they are having, because they're under continuous control and a number of them have been closed down in the last couple of years. And obviously, we've seen with them the possibility of using our technology also for food products. As I said, we're using a blend of gases with a very high level of ozone in it as an oxidizing agent for degrading the mycotoxin. We haven't found, as I said before, any residues in the food, and obviously goes back in the air through a system as oxygen without any form of pollution. We have been designing a number of plants. I will show you one of them, which goes from 6,000 tons a year, which is our smallest plant, to 72,000 tons a year of the treatable material which has contaminated. But is modular, the design is ours, obviously the intellectual property is ours, so we are also very much able to adapt to specific client requirement. We have an average cycle of less than 24 hours to achieve the results that I said before, but we are working very hard to continue to decrease the time of processing which is required, which obviously depends also on the level of contamination. This is the design of the plant for 120 tons, and the plant works, as you may see from, even if it's a very small image, based on a number of reactors where the maize is treated by per batches, and with, as I said before, a blend of gases with a dominant part of ozone, which due to the structure of the silos and due to the nature of the blend, allows that process to take place. As I said, we have been conducting several tests in Italy during the last 5 years on both Kenyan and Italian origin contaminated maize, before and after treatment, with the result that I mentioned before. We're also engaging for a further planned partnership with Ispa, which has been also very useful in the past in reviewing some of our performances. Since in 2019 we have been initially proposed and eventually we got awarded by UNIDO as the best agribusiness solution for 2019, and Ispa was involved in the review of our performances. Finally, I would say that we are also currently exploring the effect of our ozone treatment on a number of nuts. We are back on hazelnut where we started from, because there's been some requests, and also we're very interested on peanuts, where obviously there is also a number of questions that we are not able to respond right now, because we don't know how the fatty acid, for example, respond to the ozone oxidization, and a number of other questions in terms of quality of the product, in terms of palatability and performance in terms of nutrients in that sense. I would just finish saying that for what has been concerning the maize, we haven't found any parameter which was going out of the range allowed by the law in Italy. And in Kenya, the law is slightly different because the maximum threshold in terms of ppb for human consumption is 10 ppb, while for livestock consumption is not yet defined, but they are orienting their decision around the 20 ppb. I mean, that's the current discussion around the 20 ppb, which will be much higher than what we produce. We have a very interesting mixed team with very different competencies from agriculture, biology, economics, and obviously engineering. As I said, we've been awarded by UNIDO the prize I was mentioning before. We're also developing pretty strong partnership with financing partner, including the Italian SACE and the IFC in Kenya. I've just put the four major directors in the team, we're all Italians, which is a bit of a strange situation. And here we have our director of partnership that can be a contact for any specific question you may be interested or any potential idea of working together. I would like just to conclude, Antonio, not only in thanking you for thanking you and the organizer, as I said at the beginning, in terms of partnership and opportunity to engage, but also the super interesting conference where we are discovering a lot of interesting possible options to work with. And it's a very interesting deck of partners and possible interaction. Thank you.
Thank you very much. Thank you very much, Luca. There is a question.
There is a question from Franz Verstraete. Is there already an application for authorizing the use of the decontamination procedure with ozone? This is required under European law.
Yes, it's a very correct question. We are very much aware of it. In fact, we are currently not operating in Europe, although we've been operating in the R&D part, where we have a pilot plant in Avellino. But in terms of the usage of ozone-treated material for food, we will start very soon a process to get the procedure happening. It's very different in Kenya, where we don't have yet a process, but we are much further engaged in that discussion. And for livestock feed is accepted the usage of such treated material, rightly so. And also we are looking with a lot of interest in the US, where in fact is allowed the use of ozone for human consumption. So there is a number of options there. The good thing is that AflaZero is a global company, so we are very open to various spaces, but we want obviously to come back to Europe and do a number of activities in Europe, where we are pretty convinced that we will make through the difficult process of getting authorized to use it.
Okay, thank you. There is another question from David Miller. Is this legal?
As I said, I mean, it's obviously it's illegal because we are not doing nothing illegal. And by the way, the quality that comes out is far much better than the thing that the people are using. But it's legal what we are doing in Kenya. It's legal doing research and development. We haven't promoted any use of any product outside of research and development in Europe, so we're not doing anything illegal. But obviously, it requires that procedure that was requested before to make sure that the product can be treated.
I think that David Miller wants to clarify the use of ozone. Yes, it's legal, but not to degrade mycotoxins. He wants to clarify this.
No, sure, but that's I mean, that's what I'm saying. It depends. I mean, it's a new research, so it's a new development. We haven't gone through yet the process of getting authorized to do it. We did it in collaboration with the University of Salerno, and we did it in collaboration with a number of private center of research. So what we have done up to now is definitely legal from the research viewpoint. It's not yet legal in terms of massive usage in Europe, but legal is a big word in the sense globally, worldwide, makes not much sense in the sense that it's not legal in Europe, it is legal in the US. So it depends.
Okay, thank you very much, Luca. I hope that between AlphaZero and AlphaSafe and all this new approach, we can really at least mitigate a lot the problems of aflatoxin contamination in Africa. Then, thank you very much, Luca. So I move again to next speaker that is Roberto Bassi. Now is connected, and let's hope everything will be fine. I repeat the title of this talk: Adapting Step Change to Control a Broad Range of Fusarium species. Roberto, we cannot hear you. We can see your. Okay, unfortunately, still we have problem with the audio with Roberto, and then we move to Annelies Müller from Austria, representing Biomin, and she will talk about Biomin contribution to mycotoxin risk mitigation. Annalisa, if you are connected, please, you can start.
Can you hear me?
Yes, we can.
Okay, and can you see my slides too?
Yes, if you put him in, okay, great. Can you put full screen? Okay, perfect. You can go.
Okay, thank you. So thanks for the opportunity to give a short presentation here. My name is Annelies Müller, and I'm working with Biomin. And I want to give a very general overview of Biomin's contribution to mycotoxin risk mitigation. So those are the 4 points I want to cover. It's first research and development, then 2 analysis methods that we offer, the information we provide on mycotoxin occurrence, and the detoxification of mycotoxins. Here I just really want to shortly emphasize that we have a strong focus on research and development and are in lots of cooperations with universities and research institutes. And to the analysis methods, I want to introduce you to Spectrum Top 50 and Spectrum 380 method. They are both based on liquid chromatography coupled to mass spectrometry. They can detect multiple toxins in one run in different commodities. And the commercial method is the Spectrum Top 50, which is done by was developed and is done by our sister company Roma Labs. They can detect over 50 different mycotoxins and metabolites, including the frequently known ones and well-known ones, and the masked mycotoxins, and also the emerging mycotoxins. The Spectrum 380 method is not commercial and is conducted at the University of Natural Resources and Life Sciences. They have a department in Turin and we have a good cooperation. And with this method, they can detect over 700 different mycotoxins and other fungal metabolites in one run. They can also detect bacterial and plant toxins, and on request, also pesticides and veterinary drug residues, so to get really the full toxic load an animal could face in the field. If you're interested in this method, there was just a recent publication by David Steiner, who was at the university. We provide a lot of information on occurrence of mycotoxins. So the Biomin mycotoxin survey is well known. It started in 2004 and a lot of samples of raw commodities, but also finished feed that are mostly intended for animal consumption. So feed for animals, it's incorporated in the survey. And then we try to do those risk assessment maps. Here you can see the results from January to June 2020. Unfortunately, the data up to September, it's not yet processed. So we indicate the risk of mycotoxin contamination in animal feed with the colors. In red, it's an extreme risk, going down to a moderate risk in yellow. And this is based on the average contamination and prevalence of aflatoxin, cerealenone, deoxynivalenol, T2 toxin, flumonisins, and ochratoxin A. As we have the survey for a long time, we can also look at 10-year trends, for example. I don't want to go in too much detail, but in orange you see the prevalence, so the percentage of positive samples, and in gray with the red dots, the average contamination in ppb. And just for example, for deoxynivalenol, within the last 10 years, we see that in all of the years, So please forgive me, this is for all samples worldwide, but focusing on corn, and that in all years, the oxynivalenol was present in more than 60% of the samples. Similarly, for fumonisin, we see they were present in over 70% of all samples throughout the last 10 years. What we also can observe is the co-occurrence of mycotoxins. Here I want to present you samples that have been tested for at least 3 mycotoxins. It's between January and August 2020, and we can see that in 69% of those samples, more than 1 mycotoxin could be found. When we are now looking at the results from the Spectrum 380 method, this is the one that can detect over 700 mycotoxins and fungal metabolites. We can see that in 30% of the samples, 40 to 49 metabolites were detected, and only in 1% of the samples, less than 10 metabolites. Since a couple of years, we have now also focused on emerging mycotoxins, and as those are covered by the Spectrum Top 50 method, which was introduced in 2018, we have now a quite good data set already. With emerging mycotoxins, I want to refer to those mycotoxins that are not regulated and not tested on a routine basis, but are frequently found already. And here you can see from January to September 2020, the most prevalent mycotoxin found was deoxynivalenol in 61% of all samples, so all commodities and worldwide. Equal with fumonisin B1, but then already we see molybdenum, and a bit further down with 49% ovaricin, altenariol, and also the emiatins are quite frequently found. Such occurrence was also published in 3 publications last year, and 2 of them, they also tested the cytotoxic effects of the emerging mycotoxins found in vitro assays. Yeah, so the main point was that emerging mycotoxins are really frequently found. Something new we have to assess the mycotoxin risk. It's the BioImmune mycotoxin prediction tool. It can, on a global scale, predict the risk of contamination with aflatoxin, deoxynivalenol, fumonisin, and cerealenone in corn, and for deoxynivalenol and cerealenone in wheat. This is based on a lot of different models, on plant growth model, fungi growth models, on the weather forecast, on hourly weather data that is included, and it's incorporating mechanistic models of Professor Paolo Battilani and was also developed in collaboration. And those models have then been adapted with our BioImmune mycotoxin survey results and linked to global weather data and crop data. So we really hope that this helps to adapt maybe quality control and to know the risk in advance in the different regions. Finally, just briefly, I want to come to our detoxification strategy, which is MycoFix. Its aim is to reduce the mycotoxins in animal feed. It's based on adsorption for mycotoxins that can be adsorbed. This is due to the minerals included. It's authorized by an EU regulation that it can detoxify aflatoxins in the feed by binding. It can also bind ergot alkaloids and endotoxins, as we have seen in trials. And then there are a lot of mycotoxins that cannot be bound, for which we use the biotransformation with specific enzymes and biological components which convert the mycotoxins in the digestive tract of the animals. Additionally, we want to use a bioprotection which supports the liver and immune system and the intestinal integrity. For the biotransformation, which is a big focus in our company, we have the BioImmune BBSH797, a live strain that is authorized for swine and poultry in the EU, and it can detoxify trichotizines. Then we have the purified enzyme FumZyme, which can detoxify fumonisins. And now in progress is a new enzyme, Senzyme. It will be also purified and can detoxify cerealenone. all those biotransformations are irreversible and result in non-toxic metabolites. So this was a very rough overview. I'm already at the end and want to thank you for your attention.
Thank you very much, Annalise. There are no. Can you put on your camera? Anyway, there is no question. Okay.
Sorry.
Fine.
No
problem. At least we can see you now. So there is no question from audience, but I have a question. You mentioned a study about the combined toxicity of DON and the so-called emerging mycotoxins. I suppose, let's say the ones which are not regulated, Bovaris, Inanitis, Mononiforme. And you mentioned some combined toxicity on pigs' intestinal lines. Do you have more details about this combined toxicity? Because I think this can be an important issue, because these emerging mycotoxins are often occurring in the food commodities.
Yes, so we test quite a couple of them, and to be honest, I have to look in the publication. I think for some we saw a bit increased effect on the intestinal, on the epithelial intestinal cells that were tested.
Okay. Okay, is done? Okay, thank you very much. We need to move to the next speaker. Thank you, Annalise, very much. And I'll give now the words to Olga Averkieva from the company Adisseo, France. She will tell us about Adisseo, a global leader in animal nutrition with a comprehensive range of feed additives and services.
Good afternoon, Europe. Good morning, Americas, and good evening, Asia. I hope that you can hear me well and you can see my screen.
Yes.
Good. Then I would like to introduce Adisseo name. It's quite new name in the mycotoxin world, but this name still contains more than 20 years of experience in mycotoxin management, and I will explain next slides why. Adisseo is a part of ChemChina Blue Star Animal Nutrition and Health Division. It's a small part of the very big company, which is including several other chemical facilities busy in chemical industry as well as in food and feed industry. Adisseo experienced several acquisitions last years, and one of the latest was 2018 acquisition of Nutriad, and this name is more known in mycotoxin world. And by this acquisition, of course, Adisseo became a player, a significant player on the market of mycotoxin deactivators and binders. Maybe you also heard that this year we acquired also Framelco company, which is also dealing somehow in mycotoxin binders in some regions of the world. We have quite a big team in Adisseo. We have more than 2,000 employees, and we are proud to have more R&D centers and coming more and more in years. And currently we are having 10 different R&D centers located globally with concentration in France. And we have a lot of patents and researchers working in cooperation with Adisseo company. We are active in business in more than 100 countries, and currently we have a turnover of about 1.5 billion euros. Adiseo has different platforms, different categories. Probably you know the name was beginning with vitamins and methionine, but later some other products like probiotics, selenium, organic selenium sources were added to the Adiseo portfolio, as well as the full range of products from Nutriat, including mycotoxin management. Mycotoxin management of Adiseo is not only products. This is a combination of products and services. And services, in fact, we have more than products themselves. And our services are called MycoMan, and they include different types of services offered to customers. This can be MycoMan Predict, which is early prediction models before grain is harvested. MycoMan Harvest Bulletin, which is assessment of harvest during harvest or right after the harvest of corn and wheat. MycoMan Test, quick test. This is a specific ELISA-based or immunoaffinity immunobodies test-based quick tests, which are available for customers for feed mills or animal producers. We have MycoMan test in a laboratory, certified and accredited laboratory using LC-MS/MS technologies for testing more than 15 mycotoxins in one run. We have the most used MycoMan mobile app, the most used risk assessment app in the world related to mycotoxin risk management and risk assessment. This is specific to Adiseo company and also offering customers a range of our products and dosages according to the customer situation. And last and not least, we have 3 famous brands of products, Unique Plus, Toxinil Plus, and Toxinil, which are binders or deactivators, depending on the positioning. Recently, we launched, it was not a big launch, but still this year in several countries in Europe, we launched MycoMan Predict, a unique tool in cooperation with Syngenta. And I hope Syngenta will have a chance to speak and will fix an issue with microphone today, but it will be a slightly something else. But again, Syngenta was our partner in development of this service tool, MycoMan Predict. And we are using their mathematical models called Qualimeter for corn and wheat, the most accurate in the world for quantitative prediction of mycotoxin risk in 2 crops before the harvest. And this model includes climatic conditions. Of course, weather is a key factor, but not only. Agronomic practices in each country and sometimes in each field of the country or each region of country is different. And Syngenta and MycoMan Predict, they take this into account. And this is based on many years of research of Syngenta and the big data and very high correlation. But for instance, here on this slide, I'm mentioning that the model for France for wheat and deoxynivalenol prediction, the correlation is above 0.9. So it's a very high correlation. So before the harvest, around 2 weeks or even before 1 month before the harvest, we are able to say what will be the levels of deoxynivalenol, not only occurrence, but also levels. And finally, product brands. There are 3, but the maximum, the premium product, has 5 modes of actions, and others have less modes of actions. Those products are not only targeting mycotoxins, but they also target their effects, secondary effects, and repair some damaged metabolic paths and organs. These products were tested in vivo. So we believe in vivo results more than in vitro results. And in more than 40 animal trials, we could mention and we can prove that our products are working against different mycotoxins, but also in different species. And for different species and even subspecies or categories of animals, we have different recommendations and different risk estimation according to age, according to mycotoxin presence, and based on that, our MyCommand mobile app can offer the best product at the best dosage in order to get the highest return on investment for mycotoxin risk management strategy. I would like to thank you all participants, and I wish you a good conference further on.
Thank you, Olga, for the very nice presentation. I have just a curiosity. Which among the species that you have as a target is the one where the products work well, better? I mean, there is some species which has a better performance on the absorption and the inactivation.
Yeah, for the absorption, we can target some mycotoxins like aflatoxins or ergot alkaloids, and any of our products can work well. It depends on other challenges as well. If aflatoxins and ergots are also combined with deoxynivalenol or fumonizins, that of course we need to focus on other strategies, other modes of actions, not only adsorption, but also protection from some damages. That, for instance, it is known that deoxynivalenol is very rapidly absorbed in pigs, in stomach and small intestine, very fast. It does not reach a large intestine or distal parts of small intestine. It goes to the liver, and it can destroy liver there somehow, or if liver is challenged by other means, by other toxicants or antibiotics or any other stresses, we need a product which can help liver to recover, but also to enhance the liver abilities, natural ability to detoxify deoxynivalenol, which is a normal case for healthy pig. 100% of them can be detoxified by liver.
Okay. Thank you very much for the answer. Thank you very much for your presentation. There is a question, quickly, Alessandro.
From Rebecca Guimet. She asked, with MyCommand, do you give growers recommendation for irrigation if drought may be contributing to aflatoxin contamination?
Do you mean the MyCommand Predict tool or the MyCommand mobile app?
Do you give growers recommendation for irrigation? Predict or
for
out?
irrigation?
Predict or out? Both.
We normally don't target customers who are producers of the grain, but the question is very relevant because irrigation is a key point for aflatoxin contamination of maize. And what we have seen already in some countries that the aflatoxin risk this year can be associated with the irrigation or non-irrigation practices. We cannot, as ADCO, we do not have grain producers as our customers. We are dealing with feed and animal industry, but our partner Syngenta for sure notices that the key thing, this key parameter is irrigation, and for sure tells grain producers where they can reduce risk.
Thank you very much. So it's time to go forward, and we'll try again with Roberto Bassi from Italy, from Syngenta company. And let's hope that his talk on adapidine will be now possible. So Roberto, can you try to speak? No, I'm sorry. Roberto, there is a problem with your microphone because even if you talk, there is no connection. So let's see later. We try now to recover the talk of Dimitrios Drakopoulos from Switzerland. He was talking about innovative cropping system and biopesticides to prevent mycotoxins in wheat. So Please, Dimitrios, if you can start from the last slide you were talking about. We don't hear your
can
voice.
you hear me?
Now we can hear your
Can
voice.
you hear me?
Okay, go ahead.
And just 1 second. Can you see the screen?
Sure, you can go on.
Okay, thanks again, and I hope it works this time. I changed laptop, I changed everything. So, and thanks a lot to you and to Alessandra for organizing this very nice conference. So I will go back to the prevention strategies and how we can control Fusarium head blight and reduce mycotoxins using innovative cropping systems and plant-based biopesticides. So here we can see 3 prevention strategies that I examined during my PhD work. This was the cut and carry biofumigation and use of botanicals, the maize intercropping and cover cropping in order to suppress Fusarium inoculum and reduce mycotoxin contamination in wheat. We also try to directly control Fusarium head blight by using mustard-based botanicals, but today I will focus on the findings in the prevention strategies. So I will go to the first project, that was the cut and carry biofumigation and use of botanicals. For this experiment, we simulated a maize wheat rotation under no tillage, and for this purpose, we collected maize stalks, we inoculate them with Fusarium graminearum, we use 3 fungal strains, and we spray conidia. You can see here how the maize stalks look like after the incubation. It's full of mycelium. We placed the stalks in a wheat field, and later we did our treatments. So our treatments were the cut and carry biofumigation mulch layer treatments in autumn. For this, we grew white mustard, Indian mustard, and berseem clover in separate fields. We collected the fresh biomass, and we covered completely the maize stalks. The second group of treatments was to spray botanicals, either in autumn or in spring, and we spray mustards, 2 mustard-based botanicals, and Chinese galls. This is a drone image where you can see how the experiment looked like. We had mulch layer treatments, botanical treatments, the positive control, which means that we had the infected maize stalks, but there was no treatment, and the negative controls, it's uninfected, untreated maize stalks. And we always test the effect on 2 wheat varieties. So we can see directly the results on mycotoxin DON in wheat grain. And here we can see clearly that the use of mulch layer treatments significantly reduced mycotoxin DON in wheat grain. When we see the effect of the different botanicals sprayed directly on the maize stalks, we can see also we had some significantly lower mycotoxin DON in wheat grain compared to the positive control. And again, most of our treatments significantly reduced mycotoxin Zen in wheat compared to the positive control. Why we used botanicals, and especially the mustard-based botanicals and Chinese galls? We believe that these have a great potential to be very good biopesticides against Fusarium head blight. That was a very nice collaboration with Professor Giuseppe Mecca from University of Valencia. And we found a lot of different bioactive ingredients in the mustard. These are the glycosinolate and the hydrolyzed products, the isothiocyanates. We found a lot of different phenolic acids, and also in case of Chinese galls, we found a lot of phenolic compounds which have also antimicrobial and antifungal properties. So to sum up, with the use of cut and carry biofumigation and botanicals, we could decrease mycotoxin in wheat grain, and the botanicals showed very good efficacy in vitro, but moderate efficacy under the field conditions. Regarding the maize intercropping, briefly, I will introduce the concept. So you can see on the top the conventional way of growing maize as a sole crop in the field, but we also use an example of the maize white mustard intercropping in the photo on the bottom, where we can grow mustard between the maize rows. So in general, as you can imagine, we have increased land and resource use efficiency in the field because we grow more than one genotype in the same time at the same space. We have improved biodiversity in the field. We can suppress potentially weeds when we use mustard between maize rows. And also we can have interspecific facilitation. And with this, I mean that perhaps with mustard, we can control the soil and residue-borne pathogens such as Fusarium graminearum. So what can be a disadvantage? This can be the potential interspecific competition, which would mean losses on yield. And also, what about the production costs for the farmer? So we set up an experiment in the field. We tested different intercropping systems compared to the sole maize. You can see here our different intercrops that we used. We use again mustard, red clover, Sudan grass, and Phacelia. We did no tillage or reduced tillage after we harvested the maize. And we tested the effect of these treatments on Fusarium and mycotoxins in wheat. The first big question was if our intercrops would reduce yield of grain maize, and we were happy to see that none of our intercropping treatments reduced grain yield of maize compared to the sole maize cropping. And when we see the results of on mycotoxin DON, the effects of different intercroppings, we can see that when we used maize Indian mustard or maize white mustard intercropping, we had a reduction on mycotoxin DON in wheat grain by 32 to 52% on average. However, when we repeated the experiment for the second time, we had a very high contamination with DON, and unfortunately, none of our intercropping systems reduced significantly the DON in grain. A very important aspect of these systems is to investigate what are the gross margin effects. So we found that using maize intercropping systems, the farmer will receive between 14 to 25% less turnover for his crop, or her crop. And by using reduced tillage, again, we found that if the farmer used maize intercropping systems, we'll also have between 12 and 18% less profitability for his farm compared to the sole maize. And these differences on gross margin are mainly explained because of the higher operating costs. The farmer has to pay more money to do the sowing of the intercrops as compared to the herbicide, which is lower price. So to sum up, intercropping did not reduce maize yield significantly. In some cases, maize white mustard or maize Indian mustard intercropping reduced mycotoxins in wheat, but as we saw, there are some economic trade-offs, and it was not effective under very high disease pressure. That was in the second time that we repeated the experiment. So the third and last experimental approach was to test different cover cropping systems, and this is the design. We had the silage maize as a previous crop and different interval cover crops. Again, we had white mustard, Indian mustard, and winter pea. We had a positive control, that's a field that we spray herbicide. We had no cover crop, and a negative control. We did not have any Fusarium inoculum because we plow, and therefore all the residues were below ground. We tested the effect on spring wheat using again 2 wheat varieties. Briefly, the main results, we see that by using cover crops in both wheat varieties, Digana and Fiorina, we see that we successfully reduced mycotoxin DON in wheat grain, and the results were very similar and comparable to the plow treatment. So use of cover crops seems to be a very important tool to reduce mycotoxins. However, again, we performed this gross margin analysis, and we found that by using cover cropping systems, the farmer will receive between 7 and 14% less profit. And to sum up, interval cover crops are very effective, but again, we have these economic trade-offs. So if we put all the cropping systems together against mycotoxins, we found very effective and consistent results with the use of cover crops as interval crops between maize and wheat, and especially using white mustard or Indian mustard We have very good results. The use of cut and carry biofumigation is a novel approach with mustard and bersim clover. Effective but less consistent was the intercropping system with mustard, and also the application of botanicals have a great potential, but we believe that a very effective formulation would stabilize the product, as you heard earlier, and we have to do future studies on the formulation of the botanicals as by pesticides. We should always think that the cropping systems can have additional benefits for the farmer, so they can control weeds, they can build up soil organic matter, improve biodiversity. So it's not only about the mycotoxins, but there are many other benefits. But there is a conflict between crop profitability and food safety goals. And there it comes the policies that should support farmers to use innovative cropping systems and to compensate for this yield loss and this economic loss. And therefore, with the right policies, we can also manage effectively mycotoxins. I would like to thank my group, and especially Susanne Vogelixan for being my supervisor, as well as my co-supervisor, Professor Johan Six from ETH, and all the collaborators of this project. And thank you very much for listening.
Thank you very much, Dimitrios. Finally, we did it. It was a long trial, and then we did it. I have just a quick question for you. If you have to talk with the farmer and to suggest him some of the different tools you have tried to reduce the mycotoxin fungi in the crops that you target, which one you will suggest more? According to your
in my opinion, yes, I thank you very much for this very nice question. So I believe that, as you said earlier, that it's not one single silver bullet. So we have to go to the farmer, we have to see the system. So the mycotoxin management in Europe is very different from the mycotoxin management in US or in China. So it really depends on the regional conditions. For example, in Switzerland, we found that use of cover crops, it's a very effective tool, and especially when the farmers use mustard or winter pea, we found very good results. So we should make a break between cereals. We should, for sure, as a beginning, farmers that continuously use cereals, they should have an interval of cover cropping. And also other tools like biocontrol agents could provide solutions. But for my data, I can say that cover crops are very effective, but it really depends on the regional conditions. So we cannot say one treatment. It's an integrated approach.
Thank you very much. So the take-home message is look always to the local situation, to the regional situation, and try to be as more integrated you can be. Okay, thank you very much, Dimitrios. Now,
thank you.
We try again to recover the talk of Roberto Bassi from Syngenta. We will overcome the technology. So Roberto, try with the voice.
Can you hear me?
Yes.
Finally,
we got it.
Right, sorry.
So now
okay,
you can share your you can share your screen, and then you can go with your presentation.
Okay, I apologize for the previous trouble. Honestly, I don't know what is wrong, but can you see my screen now?
Sure. Put it in,
okay.
Please, in presentation modality. Just clicking on the there. Perfect. You can go.
Okay. Good afternoon. I'm Roberto Bassi. I'm R&D specialist for seed development for cereal in Syngenta in South Europe. I will present the new active ingredient adipidine that should be considered a step change in control of the range of Fusarium species. Adipidine is a brand new active ingredient. It started 8 years ago, the development, having more than 9,000 different active ingredient with a certain similarity. And after the screening in vitro and in the lab, we arrived to have two compounds to be tested in greenhouse and in the field, to arrive finally after 8 years of studies and screening to the full development of ADPD. ADPD now is under approval or under registration in several several countries in the world. As you can see, the first introduction we had, it was in September 2015 in North America, and of course is in different countries actually is under registration or registered in several crops, and as well in wheat for the Fusarium head blight control. As you can see, the different progression and the different crop, the colors means the different crop that or group of crop where ADPD is registered or under registration in different part of the world. ADPD is a trademark of the name of the IUPAC name of the PD flumethophen that is belong to the category of the SDH compounds. The SDH compounds are characterized by one pyrazole amide part, one methoxy group, and one lipophilic part, that is the part that gives the persistence in under the leaf surface or the part of the vegetal surface. Is a racemic blend, and should be considered one separate chemical group from in the FRAC list. FRAC is the entity that give rules for avoid resistance in the use of active ingredient for several use. Actually is listed with the code 7 in the FRAC due to this unique chemical composition and chemical structure. As you can see in the FRAC, within among the SDHI compounds, ADPD is classified with in a separate part with the code number 7, but with the same target site C2, that is the target site of the SDHI compounds actually available in several parts of the world. We can say that ADPD is the first member of a new group, as mentioned before, because for different reasons we have a different capacity of the control for the several fungi at the different part of the plant level. But basically, this different classification is due mainly to the efficacy in terms of control of some species of fungi. I show you in detail this kind of capacity for several. This is the spectrum, the broad spectrum of the control of the different species of fungi. As you can see, ADPD is very effective in some septoria species. Is effective in some Erisife. It means the powdery mildew, as well for Mucina laricatum and Botrytis cinerea, downy mildew. Venturine equalis, apple scab, for example, and as you can see, is effective on several species of Fusarium and some kind of other fungi, the Phytophthora and someone else. Of course, we know that it's not active on obicetes like downy mildew or Rhizoptonia. As you can see in the vitro, the effect on Fusarium culmorum and Fusarium graminearum, that are the most relevant Fusarium species responsible for the Fusarium halide on wheat in the field, as you can see that there is a clear dose response, and above 1 microgram for active ingredient per liter, we are quite sure that the control of the disease is perfectly achieved. The mechanism of action is the typical from the other SGI compounds. There are no inhibition of germination, but strong inhibition of the germ-to elongation. This is the part of the most part of the efficacy on this kind of species in Fusarium. When we are talking about Fusarium, we talk about a broad range of different kind of Fusarium. As you can see, adipidine is a really step change because, as you can see, it is effective much more than the other one, SDH active ingredient of much more species of Fusarium. And this is the reason is something that is classified with a different chemical part among the FRAC classification for this particular activity on the Fusarium species. Going faster, as you can see, the benchmark of the efficacy in the field against the gray mouth on grape and the Fusarium graminearum on wheat. As you can see, adipidine is much more effective than the other one compared to the dose rate used in the field. And especially on Fusarium halide on wheat, as you can see, the efficacy of the adipidine is much more relevant compared to the other active ingredient belonging to the different chemical class, like protoconazole and metconazole, that are quite effective, but not like the adipidine, especially in as well is appreciate adipidine, the stability of the level of efficacy always achieved from this active ingredient. Look in the field performance measured in several parts of Europe. As you can see, in the field condition, the combination of adipidine and protoconazole, that is one solution to avoid resistance, combining two different modes of action, product activity with different mode of action, achieve the best efficacy in the quite high level of disease observed occurring in the field. Considering again the instruction and the guideline for the use of this new active ingredient on Fusarium halide, but also as well in other disease, of course, we have to take in account a different strategy. That's the fungicide program must be effective in disease management. The number of application of the same chemical class of the SDCI is fungicide is should be limited according to the crop and according to the disease. And of course, the mixture, as mentioned before, with the fungicide, a different mode of action is always welcome in order to avoid the problem in resistance raising from the fungi. And another relevant part that's all this kind of fungicide should be used as preventively, not curative, in order to decrease the probability of the resistance induction. Of course, chemical like adipidine is just one brick of the overall strategy where we have to consider, as mentioned before, than the speaker, the previous speaker. That is a combination of different parts, and we need to understand all this part for a complete strategy against Fusarium halide in the field. This is another other trial that we carried out inside the MycoKey project of Fusarium halide. carried out in two different parts of Italy. On durum wheat, that is the species much more sensitive to the Fusarium red blight compared to the bread wheat. As you can see that the combination of the dipidine and protoconazole achieved a very good result in terms of control of Fusarium red blight in the field. Much more than the other solution, combination of two different triazole. And you can see as well the stability of this kind of control achieved in the field. This is the effect on the toxin production caused by the Fusarium red blight attack. As you can see, with the solution of Syngenta strategy, we are that means that we have a combination of two different active ingredients in the different part of the growing cycle. We were able to achieve the lowest content in the deoxyvalenol, considering that the threshold by law is 1.7 ppb. And with the chemical application, we are able to keep under control this the deoxynivalenol production caused by the Fusarium red blight attack, much more than treated naturally with natural infection and with artificial infection, of course. So at the end, this result give us the good opportunity to remind that this new chemical is very effective and is a good solution to bring another innovation in Fusarium red blight control in wheat and in other crop where are potential users for the food chain, especially in this case in wheat for all the purpose for the food industry.
Thank you,
So
Roberto.
That's all. Any question?
Thank you, Roberto. There is a question, actually.
There is a question from Susan. I would like to know if they you tested the effect of Fusarium POE also in the field, if this is an emerging in several regions due to the climate change as well. And also could be checked by measuring nivalenol instead of only deoxynivalenol.
Yeah, sure. These all the studies are ongoing because we know that the toxin nivalenol and Fusarium pulmora or nivale are not the only one. We had several studies ongoing on this point, and for sure, at the end, the reduction of all the toxin managed with a proper chemical management with adipidine is much better than the actual standard active ingredient standard actually we got. But of course, the study is ongoing, and we have to finalize as soon as possible to give also this information as well.
Thank you very much, Roberto. Nice presentation, nice answers, and I thank to all the speakers of the session and also Dimitrios that join us also now. We go toward the closing of the day with the talk of Jean-Loup Dorn from Italy. Actually, Jean-Loup should be French, but he works at the European Food Safety Agency. He will have a talk on risk assessment of combined exposure to multiple chemicals, EFSA from guidance documents to applications. So Jean-Loup, if you are online, you are ready for presentation, you can start to talk.
Can you hear me okay?
Yes, we can hear you perfectly.
Okay, great, thank you. Let me just share my screen.
Great, you did it.
Can you see? Yeah?
Yes, perfect.
Okay, so yeah, before I start, I would like to thank all the organizers of the MycoKey conference for inviting me. It's a great honor to present the work of EFSA in the context of the work we do on risk assessment of mixtures or so-called combined exposure to multiple chemicals. I've been working at EFSA for the last 14 years, and been mostly involved in developing methodologies for risk assessment and a number of open source databases. So today I will not only focus on the mixtures, but also on the tools that we can use to actually address this issue in practice. So first of all, I just wanted to start with this cartoon that I like a lot, what I call the nightmare of the risk analyst. We have risk perception from the public. We have the risk analysis, which is, of course, including risk assessment. If we do the wrong measure, then the risk manager might take the wrong decision. And here we have an example. So really our work is to make sure that we answer the question from the risk manager, which is a very important part of the work. In this context, I just wanted to give you a little background of the way we function in EFSA. So we basically get a question from the European Commission or from the European Parliament, from member states, or if we are masochistic enough, as we are in our unit, the Scientific Committee and Emerging Risks Unit, we create the question for ourselves because we've identified an issue to solve. So we do get, we formulate the issue with a question. We then produce terms of reference and background to contextualize it. The output is a scientific opinion or a guidance document, which has implications for risk management and, of course, for risk communication, because EFSA's remit is to do risk assessment and risk communication. The risk management being done at the European Commission, Parliament, and with member states. And of course, the risk communication then involves a number of stakeholders, such as consumers, media, industry, and professionals. A key aspect of what we do is transparency in risk assessment. So we got involved over the last 4 or 5 years to develop guidance documents, for example, on weight of evidence, which is illustrated here, which is extremely important for us, in relation to also biological relevance of toxicity or adverse effects, biological hazards, and so on, and also uncertainty. Of course, weight of evidence and uncertainty are very related. A couple of years ago, we had this conference, the third EFSA conference, and a famous statistician called Professor David Spiegelhalter from the University of Cambridge discussed the fact that organizations should not aim at increasing trust, but rather aim to demonstrate trustworthiness. And the information we produce in the context of EFSA for our risk assessment should be accessible, intelligible, usable, and accessible. So I will illustrate that further in my talk on how we try to address this important issue, not only for public health agency, industry, stakeholders, and of course the public. So before I go on to mixtures, I just want to contextualize how we do risk assessment in a very, very basic way, just give you the very basic, so that what I talk about later is kind of introduced. So risk assessment, we always have to ask a question. So we try to make the risk assessment fit for purpose. We use tiered approaches, which I will explain later, which basically the tier you would use would depend on what data you have, what time you have, and what are your resources. And we have, of course, those 4 steps. When we talk about chemicals, we first identify the toxic effect, and we quantify the dose response of the toxicity to derive what we call a reference point or point of departure, and of course a safe level, which is called the reference value in EFSA's vocabulary. Of course, balancing the toxicity with how much of the chemical is in the food in the context of food safety, exposure assessment, where we combine the occurrence of the chemical and the human consumption or the animal consumption or concentration in water if we're dealing with aquatic organisms. Combining the two, we get the risk characterization, which is the confrontation between the hazard and the exposure. And that gives us the risk. So again, when we go a little deeper into the biochemistry and how we try to understand how toxicity occurs, the first thing we are interested in is what the body does to the chemical, which is what we call the toxicokinetics, and what the chemical does to the body, which is what we call the toxicodynamics. And of course, if we just have an external dose and a toxicity marker, I don't know, a lethal dose, we can't do very much with that. What we're trying to do is to get to the bottom of that slide where we have the external dose, we ideally move into the internal dose to the target organ dose, target organ metabolism, target organ response, and toxicity. And if we have that quantitative continuum, we can do a proper job. So this slide illustrates how we can map the level of knowledge we have in relation to exposure, hazard, and risk. When we have, let's say we are in a tier 0, we have 1 week to do an assessment. We have just a fax from Portugal giving us 2 concentrations for mycotoxins, and we have to assess that new mycotoxins for which we have no toxicity data for breakfast cereals in children. So there we have to be a little creative because we may not have the data. We may have to use in silico models to predict the toxicity or read across between a known mycotoxin which has a similar structure and use these data. Of course, if we have a lot of data sets for quite a lot of mycotoxins we do, aflatoxin, dioxin, DOM, and so on, we can do really a proper job and look at the mechanistic aspects of the toxicity. So this is quite important because all the methodologies we can apply will depend again on the data we have and the time and resources we have. So in the light of that, when we wrote the guidance document on weight of evidence, we tried to generalize that, and we thought, okay, we have to think about when we have data, whether it's a regulated chemical, it's a dossier, it's a pre-market assessment for pesticides, for example, or whether it's a contaminant that has been in the food chain for the last 50 years, like dioxins. We need to bring this evidence together, first to be transparent, but also to be robust in the science of the way we perform the assessment. So the first thing we would do is to assemble the evidence where you would identify the relevant hazard evidence. This is for hazard assessment. And then we would weigh that evidence to see whether the different lines of evidence are reliable or not, and we would integrate the evidence and report the results. Of course, when we don't have data for that chemical, like an emerging contaminant, we may have to use, again, in silico models or read across methodologies, TTC, and so on. I won't go into details, just to illustrate. Now, from all these assessments we've performed over the last 18 years, EFSA was created in 2002. We started publishing, I think, in 2003. We've evaluated roughly about 5,000 substances, and over the last 11 years, I've been involved in working on this database with quite a number of collaborators to basically put all the data, all the hazard data we've used in our risk assessment since EFSA's creation in 2002. It's useful for us because if we have a crisis for a known hazard, we can access the data fairly quickly, and it can be a useful tool for stakeholders. For example, a number of agencies have been using it. CEFIC has been developing a tool called AMBIT2, which brings ECHA's database and EFSA's database for read across, for example, to try to predict toxicity. It will, of course, avoid the duplication of efforts across agencies, and it provides a basis for international harmonization and data sharing, because the data is structured using the OECD harmonized templates. So very briefly, what does it contain? It contains chemical information, the information about the EFSA output, and of course the basic toxicological information. You can access all the data in the database. This is the latest version from the 27th of March, and you can see it's been nearly 5,000 downloads and over nearly 8,000 views. So people are using the database. We're very pleased. And we also have a MicroStrategy tool which allows you to search hazard data for specific compounds. And if you're interested in species, colleagues in the data unit have developed this tool where you can download, for example, all the NOAEL values for the rat, for the humans, or for the dog, for the mouse. You can see that the rat, of course, the rat data is the largest section of the database. We also have that for the ecotox, of course, bees, fish, and so on. Well, it's nice to have a database, but we need to always update the database and start to include new properties. So we thought, well, for the time being, we ought to include physicochemical properties, toxicokinetic data, bioaccumulation data, the summary of our exposure assessment, exposure estimates, and more mechanistic data, because if we understand the mechanisms, it will help us to provide better advice in terms of public health, animal health, or environmental risk assessment. Of course, we would like to formalize the way we perform the weight of evidence for transparency, but also the inclusion of biological relevance and uncertainty, and work on this mechanistic template from the OECD, which is called OHT 201, and the toxicokinetic template, which is called OHT 58, because we can then structure these data sets and again inform better our risk assessment. The third aspect is to link this, say, fixed database in a more dynamic way with modeling platforms, and we're working rather a lot with Professor Emilio Benfenati in Milan, who has developed the Vega tool, him and his team, and we're trying to connect those models to the Vega tool, so that we may be able to compare published EFSA values and predicted values. And when we don't have ecotox data, say for a food additive, we might be able to provide some predictions for that without testing. So this is what it will look like. We would have the chem information, the exposure, the EFSA output, the tox information, and it will be linked to a modeling platform. All of this to start implementing what we call new approach methodologies, so-called NAMs, in risk assessment, start using alternative to animal testing and in silico tools and so on. These are examples of the QSAR models we've published recently. There's a NOAEL predictor. There is a predictor of acute toxicity in fish. And from some quite complex meta-analysis we've performed on toxicity of plant protection products and veterinary drugs in bees, we've then developed a first QSAR model to predict toxicity of mixtures. So the heart of my talk is really looking at the assessment of multiple chemicals, so-called mixtures. And here you have the example of a cocktail effect in Parma, where EFSA is based. We do like food and we do like our digestive, but if you mix a limoncello, a barniolino, and a nocino, which probably contains the highest amount of alcohol, you are nearly guaranteed a headache in the morning. That's what we would call a practical cocktail effect. So joke apart, last year we published the Mixed Tox Guidance document, which is a harmonized guidance dealing with human health, animal health, and ecological risk assessment of those mixtures. The idea is that we may have to deal with a whole mixture, for example, petroleum in the sea. That would be a whole mixture. We may take the whole effluent. Or we may have to deal with a group of pesticides, a group of contaminants, a group of feed additives for either the pig, humans, or the bee. And we may have to deal with interactions. If they interact together and become more toxic, we have to take into account in our methodologies this type of interaction to be protective. Of course, the first step would be the problem formulation, where you define your question. We then have the exposure and hazard assessment, and then we combine all of this exposure and hazard assessment for risk characterization. Interestingly, it's very important to be able to report the summary of the assessment, again, for transparency, but also for other scientists to replicate it if needed, or as example to illustrate the use of our methodologies. So this is what the guidance, the content of the guidance. So I've just been through it. We've harmonized the chapters with general consideration, whole mixture approach, component-based approach, and of course the Turing principles. A number of case studies, one for human on some contaminants, animal health, the risk assessment of substances in essential oils used as feed additives, and how do we assess synergism in bees for pesticide risk assessment or any other assessment of contaminants. So the harmonized framework is presented here. And again, I won't go into too much detail because I have some slides for all of those steps. You have the problem formulation, you have the hazard assessment, you have your exposure assessment, and the risk characterization. There are a number of factors influencing each of those steps. The assessment sequence, dose addition is the default model. All the chemicals combined together are additive, unless we have evidence for interactions. And we may have to bridge data gaps, because if we have a large group of chemicals, we may not have the toxicity data for all of them. So that's an important aspect of the work, is to basically bridge those data gaps. So let's go into the tiering principles. Again, when you're in a data poor situation, you will be further away from reality. When you have a lot of data, you will be closer, you will decrease the uncertainty, the accuracy will go up. Again, the relationship between the tiers, the data availability, uncertainty, accuracy, and the outcome of the risk assessment is key to the tiering principles. So I would leave it at that. The less data you have, the more you would have to use default values or models. The more data you have, the more complex your assessment can be, and the more precise, I would say. So let's talk about the problem formulation. So basically, the first question you have is the risk assessment question. Are we dealing with humans? Are we dealing with adults? Are we dealing with children, farm animals, a species of environmental relevance, the whole ecosystem? What is the question to assess, and what is the mixture we need to deal with? We then, how do we deal with this risk assessment with a conceptual model? Where does it come from? Is it in water? Is it in food? Is it in, what are the exposure pathways, species, subpopulation? Is there a regulatory framework that will really structure the assessment in the first place based on the legislation? What is the methodology we're using? We make an analysis plan, and then we go ahead and perform the risk assessment. Of course, it's an iterative process. We may have to update and modify this conceptual model, analysis plan methodology, and conceptual model. The same applies to the exposure assessment. And the very basic question is, how much of the chemical is in the food or in water? How much of that food is consumed? And then we combine them together for the exposure assessment. You may have a lot of occurrence data, but the consumption data may be poor. So to note that the occurrence and consumption tiers often do not match, and the resulting exposure tier will be determined by, again, the available data, including for the occurrence of different components of a mixture. We may have it for 5 compounds, we have to deal with 10. So again, we have to report all of this. And of course, we go from default value in a low tier to really precise as probabilistic risk exposure assessment when we have individual co-occurrence data. And I will provide an example later on. When it comes to the hazard assessment, basically, we need to understand the toxicity of the chemical. We just need to collect these toxicity values, and we need to understand whether the dose addition applies or if they interact together and become more toxic. That's the bottom line. So we basically summarize all of that into a table. And for risk characterization, we, of course, combine the exposure metrics and the hazard metrics, so the exposure and the toxicity data. I just have a little issue because I cannot see the full slide. Excuse me. Oh, I've done something wrong. Okay. So basically, once we combine the exposure and the hazard metrics, we would confirm the approach for the risk characterization. We have developed a range and reported a range of methodologies. We would summarize the results and interpret the results. Of course, if the combined risk is acceptable, we stop there. If it's not, we may have to discuss with risk manager and maybe refine the risk assessment to a higher tier, or ask the risk manager to basically act and decrease the exposure, for example. So here is the reporting table. In the interest of time, I will not go into details, but again, it's structured for the problem formulation, the exposure assessment, the hazard assessment, and the risk characterization. So my talk was talking from guidance document to applications. So the remaining slides will illustrate a number of applications of this document in practice. The first applications, we published those reports in February, where we try to implement the mixed-tox guidance for human risk assessment of multiple chemicals, and also for animal health risk assessment of multiple chemicals in essential oil for farm animals, with some recommendations on how to improve the the methodologies, but also to develop biologically based models, which I will touch upon in a few slides. Beginning of this year, we published the results of a large project, which was called MyChief, collaboration between Piacenza, Professor Paola Battilani, Chiara Dall'Asta from Parma, University of Belfast, and I will miss a few, the INRA Toulouse with Isabelle Oswald, so quite a lot of actors, and Portugal as well, University of Minho, where we looked at three key aspects: environmental variables influencing the production of mycotoxins, of course, the influence of climate change, temperature, season, and so on, and comparative toxicity and kinetics in farm animals and humans. Just to illustrate, in terms of mixtures, we looked at a quite large database combining literature and EFSA database on co-occurrence of mycotoxins. And of course, here we have an example with this recently published paper in Microorganisms. So we're looking at collecting of the hazard data for each mycotoxins using the open food tox database I just described. From the co-occurrence and the consumption patterns, we can actually combine that and use those addition as the default if we have no evidence for interactions. And we could use margin of exposure for the combined exposure to those multiple mycotoxins. And this can be performed for humans and farm animals. And we have a number of manuscripts that are in preparation on these applications. The full report is published since the 4th of December 2019. And this report is associated with a number of tools to perform probabilistic exposure assessment, and so on and so on. I did forget ISS, who was also heavily involved in this work. In relation to that, the FAO WHO basically funded a very interesting study, which I think is very relevant to this conference. Total diet study in 4 African countries for the first time: Mali, Benin, Cameroon, and Nigeria. And this included over 500 mycotoxins and their metabolites. So we foresee that we could perform future risk characterization for multiple mycotoxins and compare European patterns and African patterns using the methodologies we've developed in mixed tox. An important aspect of dealing with mixtures is to look at interactions. And I wanted to just illustrate that for metabolic interactions, because we do have some examples. So when we deal with, of course, mixture toxicity, and we see that there is an interaction, we need to check what it means and what is the magnitude of that interaction at the current level of exposure. Very often the level of exposure is too little, but there are some examples when it could occur, particularly in the environment. So we would then integrate that in the risk characterization, either through an extra uncertainty factor or running a biological base model, which would allow you to quantify that in the risk characterization itself. It's a bit more sophisticated, but I think we are moving increasingly towards the use of such models, because they are species specific. So you have food chemical A, external dose, internal dose, target organ may be excreted or may be metabolized, and the chemical B could block the metabolism, inhibition, or induce the metabolism, so faster elimination. Of course, the result could be, it depends if the chemical is bioactivated or detoxified, and that gives us already 4 possibilities. If you block metabolism when it's bioactivated, you would reduce toxicity, and if you block detoxification, you could actually increase toxicity. So you could potentiation, or it could result in actually a proper synergism, or you could actually decrease the toxicity through antagonism. And potentially we could integrate those magnitude in the risk assessment process. Typical example of synergistic effects is the melamine and cyanuric acid story. This is the Mackin piece in Brussels. So what happened? Melamine is used in the manufacture of plastics. It's approved as food contact material. It's been added deliberately as food adulteration to, of course, illicitly, to increase apparent content of protein, because when you use Lowry method, 480 nanometers. The fact that melamine has 3 amine groups will make you believe you have proteins. So it complexes with uric acid and it results in kidney toxicity. In 2007, there was a US crisis where pet food imported from China was adulterated with scrap melamine used to laminate tables. Actually, the scrap melamine is a mixture of melamine and cyanuric acid, and thousands of pets had severe health effects associated with a number of deaths. In September 2008, as you would know, 500,000 cases of infant children hospitalized with kidney stones from adulterated milk powder used in infant formula. And then between 2009 and 10, we reassessed, WHO and EFSA reassessed the tolerable daily intake for melamine alone for human health. Now, if we look at the toxicity of melamine and cyanuric acid, it's rather different. The mechanism is quite different. Melamine complexes with uric acid on its own, but melamine and cyanuric acid form a covalent complex, which then really blocks the kidney at doses lower than the complex between melamine and uric acid, which is actually a more labile complex, van der Waals. So melamine and cyanuric acid are covalent, so they really are synergistic, and a number of studies have been performed. And in collaboration with INERIS, we recently published the investigation of this interaction in the fish, because the fish seems to be the most sensitive species to that mixture. Another example of mixture effect, which is very, very well known, and that's why we call that the yin-yang of CYP3A4, CYP3A4 being a cytochrome P450, which is probably the quantitatively most important P450. And when you drink a glass of grapefruit juice and you take tablet, you will block the metabolism of a number of drugs, including statins, for example, and various other drugs. But when you take St. John's wort for mild depression, which is a plant we can find on the Apennine Mountains just outside Parma, you will actually do the opposite. You will induce the metabolism. And of course, at the level of exposure, we're talking milligram level of exposure for pharmaceuticals. Here we have a number of issues. So it's always written on the posology of the pharmaceutical not to drink either grapefruit juice or St. John's wort for a number of pharmaceuticals. So I'd like to talk to you a little bit about how do we refine these assessments with biologically based models. About 4 years ago, we had a conference, the URL ECVAM, which basically started to think about new generation of physiologically based kinetic models in risk assessment. We had a state-of-the-art workshop with a number of bodies, US EPA, FDA, EFSA, RIVM, Health and Safety Executive, and so on. And this resulted in actually the production of an OECD guidance document on the use of these models for risk assessment. And this document is currently about to be published, I think, in the middle of next year. So to illustrate that, we again have worked quite a lot on developing these models for species of interest to risk assessment, humans, farm animals, species of the environment. And for transparency, we always try to publish the physiological parameters from which we will develop the model and the model itself. However, this is not enough. We need to start in, here is an example on farm animals. You run your model and we need to validate the model using real data. So you would compare the observed concentrations, real measured in the animal, versus the predicted concentrations. And of course, we need to harmonize the way we report the sensitivity of those models according to the species and their physiology. For example, we can see that depending on the species, we may have, for example, body weight influencing the outcome of the model, or cardiac output, or the liver metabolism. It will depend on the species as well. So the OECD guidance document basically says that we ought to run global sensitivity analysis for each of those models as we publish them with all their parameters in a transparent way. Final example, quantitative in vitro, in vivo extrapolation. We are trying to reduce animal testing, so we're trying to use in vitro methodologies. And there are some new data requirements for pesticides, for example, fungicide. we need to compare the in vitro metabolism between the test species, which is very often the rat, and humans. We're running a number of case studies on this, and what we're trying to do again is to predict human kinetics for compounds of relevant to food safety, pesticides, contaminants, and so on. A guidance document is under preparation, and this would allow us to take into account differences in metabolism for human risk assessment, and can be also applied to mixture risk assessment, either using dose addition as the default, or we could incorporate the interactions if needed. Here is an example, collaboration with a number of partners, including ISS, University of Utrecht, and ANSES in France, where on the one side, the laboratory identifies the enzymes metabolized, and on the other side, we take viability data from the literature, mostly from the pharmaceutical database, and try to combine them to predict the kinetics of chemicals for human risk assessment. Finally, all of this work is to be integrated together with an open source platform for all the models we have for the different species to allow us to, again, combine the physiological data with the chemical specific data. And we are able to compare toxicokinetic parameters, the prediction of tissue residues. We could also perform reverse dosimetry going from the inside to the outside, for example, using biomonitoring data. And we could compare species-specific kinetics to see how they differ, and go to the integration of kinetic and dynamic modeling, of course, with the reporting of sensitivity and uncertainty. We have published an R package, which is a prototype of the database, but we're currently working on a graphical interface to make it more user-friendly. Final example, final. QSAR models developed, again, in collaboration with the group of Emilio Benfenati and Utrecht University, for risk assessment of mixtures in bees, where we look at the prediction of combined toxicity for binary mixtures, mortality data, LD50, predict whether we have dose addition or synergism, and these models are implemented in the open source Vega hub. Few concluding thoughts. Going back to the start of my talk, risk assessment ought to be fit for purpose. We use tiered approaches, and of course, the transparent reporting of how we combine the evidence together is we use the weight of evidence approach. Open Food Tox provides data for all chemicals assessed by EFSA since its creation, over 5,000 chemicals. And the Mixed Tox guidance document provides a harmonized framework to assess mixtures across chemicals, but also across species, whether we're dealing with humans, Daphnia, kingfisher, bee, or the trout. The development of this TK plate platform allows us to bring new methodologies to refine the mixture risk assessment. And of course, we always need to implement those open source tools in practice. It's not just an academic exercise, but also education. We need to train staff, experts, and the risk assessment community to use those tools. And with that, I would like to thank you.
Thank you very much, Jean-Loup. I'm really impressed by this global overview, and I think you gave us a lot of excellent stimulation. I have a question, more than a question. I think I would like to ask you a comment. According with the multi-toxin contamination of food of different from different species of the same genus, but often from different mycotoxin from different genera. Would you recommend studies about the toxicity of to increase and improve the studies that we have now available on the multi-toxin contamination from the same genus and also from different genera? I mean, we have different genera that can affect, so with different kind of mycotoxins, and some of them are not regulated. So do we really need to improve and increase this kind of study?
Excuse me, I was muted. Yeah, it's always the paradox of trying to reduce animal testing, but then you realize that we don't have any data. So it's a kind of double-edged sword. We need to generate that data. So maybe there are several ways to generate that data. The first thing is we can look at presence of genes of susceptibility in those species without testing, using comparison of DNA sequences or proteomics approaches. That would give us maybe an idea on how we could actually deal with that. But on the other hand, we may need to have to set up harmonized protocols to do in vivo studies to set the gold standards for different species. And maybe those models might be helpful to in the future to actually test the available data. I don't know if I answered your question.
Yeah, absolutely, yes. And this is really a big effort that all the scientific community must do. So I thank you very much again, and as last speaker, and I thank you all speakers. But now I would like to give to Antonio Loglieco the last words for the closing the scientific conference, the day, this second day of conference, and then I will just give you some announcement for tomorrow. So, Antonio?
Thank you very much, Antonio, for your well organized introduction about all different speakers. So, I'd like to thank all audience, all participants for these second days of MycoKey conference, online conference. And I'd like just to point out that we had so far more than 300 registration. So there's more than 300 participants, and also a high number of YouTube visualization, more than 600, 700 visualization per day. So this is, I think that this is a good, successful outcome of this conference. So lastly, I'd like to thanks to all speakers. Really, this day was very interesting. We had the different outcomes, different results from MycoKey in the prevention intervention strategy. So with a very high TRL, so very high applicable application in real life, in real field. And then also the voice from food and feed the industry was very interesting too. And then I'd like just to thank a lot Jean-Loup Dor for his really spectacular, his very interesting talk. So thank you for all, and then see you tomorrow.
Okay, so thank you very much, Antonio. Tomorrow at 10 o'clock we will start the conference. There will be the talk of Michelangelo Pascal about the 6 work package remediation, and then we will